All right, here we go. So thanks everybody for watching the stream. Hopefully we'll get a few live chatters in here, and if people also want to come on and chat a little bit, we can do that too. But I wanted to bring Nathan on, known as Pitch Lumin. That's the name of his YouTube channel. And we had a debate on Modern Day Debates a little over a week ago, and but we have talked before, and I got to know you from your first debate, which was on Modern Day Debates, and so we have debated about creation evolution. So do you want to just kind of give a brief introduction to yourself, so my audience will kind of see a little bit about who you are?
Yeah. So yeah, we had a discussion on MDD. And Chris is a very respectful guy, very polite, very knowledgeable. He's easygoing, good to talk to, and so just get along well, I feel like, and I really like the conversation. I've learned a lot from you, Chris, and so I do take the like a biblical, literal approach to my worldview. And I think that if the Bible is true, that it has very severe, in a good way, implications for humanity. That was part of like in debates, I try to lay the groundwork about why I feel that these topics are important to discuss, as far as who we are, why we're here, and so that's a little bit about my position. I really do like science, and talking with people, communication, society, technology, I think is really cool. So,
right. Yeah, and you're studying nutrition right now.
Yes. Yep. I am. I'm in school for nutrition. I have to work full time, so I can only take like a class at a time, but I'm working at my degree. And I do tend to share. I actually could do that, right? Can I just real quick share something?
Sure.
Just for so this here, I like to share this with as many people as possible. This is an angiogram, a picture of somebody's heart who had heart disease on the A side, and then the B side is a 5-year follow-up where they actually reversed their heart disease. That's what caused me to want to go back to school for nutrition. And so just sharing that with people. If you know anyone with chest pain, joint pain, high blood pressure, high cholesterol, that's photographic proof that people have had heart disease. They switched to what is a vegan, a plant-based, plant-exclusive way of eating, and they've actually been able to heal themselves from heart disease, diabetes, as well, leading killers in our country. Some cancers have been reversed. People have gotten out of medications, arthritis, autoimmune conditions. If I can, shout out to Brooke Goldner. She's a wonderful doctor. She healed herself of lupus. She goes into some really fabulous stuff with science. Y'all should check her out if you do like Brooke Goldner, VSH, it's Vegan Society of Hawaii, but she did a talk there. Very brilliant woman, very caring about people. She loves to talk to people about free. So if you have any questions, she'd probably be happy to answer. I got to shout her out because she's done so much for so many people, and that's really why I want to be in school, is to help get people if to a quality of life, health span as well as lifespan. So that's my school drive. Bible's my underlying motivation for this. My views have switched before, and where they are right now, I try to stay open-minded, and it really helps having respectful people like Chris. So
when you said switch before, what does that mean?
That would be like in regards to so like with the Earth, my view on cosmology. I've switched on that. I used to believe in like a really old Earth, the primordial soup and everything. I kind of had a view of, like, I always kind of thought Adam and Eve were the first man and woman, but I had like a hybrid view mixed in with evolution. I would say
a lot of Christians, believe that the Earth is old, that life has evolved over time, but that, maybe something happened with like the first sort of man, woman, or something to explain,
like divine intervention.
Yeah, exactly.
To give us our like consciousness or soul.
Something along those lines. Yes, right.
Yeah. And that's
if you take it literally, right, that God fashioned Adam out of dirt. And,
yeah, I was raised Catholic, and the way I
okay.
kind of understand it is like Catholics are like they believe in Christ, but they don't read their Bible or really live the Bible or anything like that. So,
Right.
that was, yeah, kind of my upbringing. I loved astronomy, and really big into space for a really long time. Took it every chance I got. Loved the PBS Nova. Really would kick myself if I missed an episode of it or something. So, yeah, that's
Your
kind of
belief in
Alex Filipenko. That dude's he's awesome.
Okay. So, but and your belief in flat Earth, like that is, I know, it's biblically rooted, right?
Yeah. Well,
Okay.
so actually,
Not entirely, right? Because you also feel you're taking an assessment of the evidence that's out there, and you've learned about the alternative model of the flat Earth and how, well, there is no gravity, right? That kind of thing.
yeah. So
Yeah.
I would say the Earth is empirically flat, planar, not moving. Based on our experimentation, and then how you interpret the results is another thing. To me, the Earth being flat does allow the Bible to have a chance of being the word of God, because God wouldn't get the cosmology completely wrong. The Bible is entirely a flat Earth book. If
Sure.
I saw definitive evidence the Earth was a globe, I would immediately disregard the Bible, because there would be no, maybe there's some poetry or something in it that we could benefit from. I do actually think Jesus Christ's teaching of unconditional love would solve all the world's problems. If some man wrote that thousands of years ago and happened to get it right, that's incredible. But I do think like forgiveness, talking with people would go a really long way, trying to work out differences with one another peacefully rather than resorting to violence. So I think that would go a long way. But yeah, if the Earth was a globe, the Bible would have no credibility at all.
Right.
So
Okay, interesting. And I think that's probably a pretty fair assessment. It's kind of obvious that the Bible is built upon the presupposition that the Earth is flat.
absolutely,
there's a firmament, and right.
Joshua asked the sun to stop moving.
But I also feel that there probably are too many things in the Bible that one should not take so literally, and that would be one of them. That's my position. But in fact, okay, so if that was demonstrated for you, then you would just be like, okay, well then therefore the Bible is not true, and you would cease being a Christian.
Yeah, because well, Jesus, I think it's kind of well documented, and I think even like a lot of like prominent atheists are like Richard Dawkins is very big in evolution, but like I think people just like Jesus existed. He was a man. If he claimed to be God, he'd either be right, crazy, or a liar or something like that. But
Right.
I think that kind of teaching has merit. But yeah, I mean, there would be no the afterlife of the Bible would not be a thing. The creator of the Bible would not be the creator, would not be the creator of reality. So there wouldn't really be a point for me to follow in that, if it happened to be, which is part of why I do take the topic very seriously.
Sure. And I think that's actually, I mean, I don't agree with you, obviously, but I do think that it's at least something to be commended, because I do think that at least you're being fully consistent with taking the Bible literally. And unlike some of your other young Earth creationist compatriots who don't believe that the Earth is flat, they just ignore those things, or I don't know what it is. I mean, it's surprising that they don't take those things as literally as you think that they might. But of course, there's an awful lot in the Bible that it's hard to take literally. And in fact, I would say, so if you could find some kind of key core contradiction in the Bible, would that be? This is not why I brought you on, but I'm kind of curious, because now we've gone down this rabbit hole a little. If there's some sort of core contradiction in the Bible, would that be problematic for you?
Yeah, it would be. there are, like, I've had some people try to have pointed some things out, and I do think like there were some kind of what we would consider like inhumane things in the Old Testament, doing maybe certain things with like animal sacrifice or whatever. But I think at the time it's what the people around God's people were doing, and so it was maybe a way to relate to the outward people, and like if they wanted to hear what God's people were doing or were interested, wanted to get involved, and it was maybe to help set people apart. So I do think that, like, I would be open to hear. I'm always open for criticism, critique. If you have anything that you think would disprove the Bible, because if the Bible, and there has been some manipulation, I think, by the Vatican over time, very corrupt organization that I'm strongly opposed to is the Vatican. But they, like, I think that the Bible is accurate, and it can kind of hold its own ground. But yeah, I mean, I'm always open to hear that kind of contradiction. And I know there are Christians out there who do not ascribe to the earth being flat or anything like that. And there was something about the debate. I could have taken my opener another way than I did, but I figured talking about like the military documents and stuff laid the premise for why it's important that we band together as humanity, rather than taking a flat earth approach. I took the people in charge are tyrannical and have no regard for our lives as people. And I cited that with the Operation Sea Spray. If you can spray your own people without asking them, and people get injured and somebody dies, it could be any one of us that was that person who died. And that's just, one of hundreds of experiments. They've done horrible stuff to our military men and women, like with Top Hat, giving them nerve agents and stuff. So that I think that laid a groundwork for why we need to use our communication, hear these networks that we have available, talk to one another and say, we don't support that kind of thing. Let's not hurt people to solve our differences. Let's talk it through. And material is material, but lives are irreplaceable. So
I want to kind of follow up on the military part that you started off with, because I mean, I don't necessarily think that it's specifically related to the topic that we were debating about, but I did have a couple of questions about it. But this is kind of a little bit of what I was kind of referring to about just internal contradictions. So this is something, I don't know if you're familiar with this, but this is Dan Barker. He is the head of the Freedom from Religion Foundation. He was a Christian preacher for many, many years, but then he became an atheist. And so he knows the Bible very well. And he has this thing called the Easter Challenge, which is basically tell him what happened on the day that is most important to the doctrine that was born. And one thing about the Easter Challenge that's kind of interesting about it is that it's one of the few places in the New Testament where we have repeated stories of the same event. And the problem with it is that, unfortunately, as you go through it, and I don't know if you can really see it because I think you're joining us on your phone, but you may want to take a look at this. But there are several discrepancies as you go through the different accounts of what happened with the resurrection. Like, when did these women visit the tomb? And who were the women that were there when they visited the tomb? It's very.
Can I ask, just is this the curious, the King James version of the Bible? This is using King James version or like an NSV or?
So it includes all of them, right? So
okay.
It's basically, they're going to be basically the same, but you have at the very early in the morning, at the rising of the sun. This is the King James version. Or when the sun had risen, which is the NRSV, or just after sunrise, which is the NIV, right?
Okay. Yeah.
They all basically say the same thing.
Mm-hmm.
But yeah, like which women were there is different across the different gospels. What was their purpose to see the tomb? They all disagree. Was the tomb open when they arrived? Matthew says no, but Mark, Luke, and John say yes. Who was at the tomb when they arrived? Matthew says that there was one angel there. Mark says that there was one young man, and then Luke says there were two men, and John says there were two angels. And where were these messengers situated? Like, there's Matthew says an angel was sitting on the stone. Mark says a young man sitting inside on the right, And Luke says there were two men standing inside, and John says there were two angels sitting on each end of the bed. What did they say? They vary in what they said. So it goes on and on. Yeah, like there's a bunch of just internal contradictions across the New Testament on this one thing that is one of the only things in the New Testament that is repeatedly told across the New Testament. So it's the only place where you have an opportunity to hear the case made from three different witnesses, or four different testimonies. And there's a little bit extra on top of that because there's some testimony that you can find in like Acts and a few other places. And it's not consistent. Like one thing is like, where did Jesus first appear to the disciples? Matthew says that it was on a mountain in Galilee. Mark says to 2 in the country, and then to 11 as they sat at meat. And in Luke, it says in Emmaus, which is about 7 miles away, at the evening, and to the rest in a room in Jerusalem later that night. And John says in a room at evening. Another really kind of surprising one is like, where did the ascension take place? Like, this is kind of important. Matthew says there was no ascension. The book just ends in a mountain in Galilee. Mark says it was in or near Jerusalem. And then Luke says it's in Bethany. And John talks about no ascension, and Paul says there's no ascension. And then Acts says that he was ascended from the Mount of Olives.
Wow, that is quite comprehensive. I will I'm gonna definitely have to check this site out. And then I'm gonna have to do a very deep study on this and try to because that is one, like, when presented with this type of information, because this is a lot. But then what I'll do is I'll go to the Bible, I'll read all the chapters in full, because I do think it's with anything you should always have the context to get a more complete understanding. And so I'm gonna look through all of that and try to figure why they might be saying different things, like with talking about Jesus showing up on the mountain. Maybe that was one disciple who saw them there, but then the next disciple later in the night didn't see Jesus until later at night and saw him in the room. So
I encourage you to take it, and then
I definitely will.
You should try to come up with do the Easter challenge, because as far as I understand, Dan Barker has he created this over 30 years ago, and he hasn't had anyone actually meet the challenge.
Wow.
Yeah. So, but I honestly, I think most Christians, as well as most people, are willing to say that, the Bible is a very old document, obviously written by people. It's inevitably going to have flaws. Most people don't accept the Bible as 100% literal. And you cannot take everything literal in the Bible. And obviously this is a case where you can't, because it's completely irreconcilable, because these things are completely irreconcilable. Like, was there one man there, or were there two men? Was there one angel there, or were there two angels? Were they inside? Were they outside? Were they standing? Were they sitting? Like, just those little details. It's like, you can't be two inside and outside at the same time.
Yeah, and sitting and standing at one time is...
Or you can't ascend from the Mount of Olives, and you can't also ascend from Bethany, and you can't also ascend from Jerusalem.
Yeah.
Right? That would be 3 ascensions.
Mm-hmm. That is something that I will definitely be looking into, because really I do feel that the Bible has to be a literal book if we want it. Like, I think everyone would agree, like, if someone died for all of our sins, we were forgiven, and we were allowed to go to heaven and live in paradise for eternity. I mean, that beats the alternative of evolution, kind of, we're here and then that's it. So, like, I think people would want the Bible to be true, and we would want that to be literally true, because we would want that. So I like people who say the Bible is metaphor, like, and there are metaphors in the Bible, but like, it's something like you can take things, literal things are there to be taken literally. And otherwise I think it kind of muddies the water of the faith. And like, I know there are people who will say, like, if you don't take Genesis literally with the firmament and everything, then why would anyone give any credibility to the whole rest of the book?
Yeah, right. And I think there's some I think that that's a fair point, and I get it. I get why some people go down the rabbit hole of creationism, because they feel that they have to justify every single part of this book, and they can't possibly allow for doubt of any part not being true, because then that might mean that the whole thing isn't true.
Yeah, exactly. Right. And I think
but
part
if
of
you operate from that position, then that means that you have to presuppose that the entire book is absolutely true, and that anything that you could see in the world that contradicts the narrative or the conclusions that one would have to draw from such a model, that the Bible is absolutely 100% true, you have to deny that evidence exists.
Well, right? I would not deny it. I would have to try to see how it could fit in, unless it is something that is definitively, like the Bible mentioned several times, that the earth is not moving. So if we detected motion to the earth, falsifies the Bible right there. And and why, like, that I think it's so important is because, like, right now in this world, we have a lot of people who are religious. What, if they're Christian, if they're Catholic, if they're or sects of Christianity, Catholicism, Islam, Judaism. There are a few like polytheistic religions and everything. And I think if these are all false, that's something that all of humanity should know, because it doesn't benefit us as a species. Taking a naturalistic approach here, it doesn't benefit our species anymore. If religion offered some comfort or ability to help create a community or society back in the past, right now we live in a time where we might have someone who is praying 5 times a day to Allah, who might be really good at science otherwise, and might be able to develop a technology that could help improve our lives, or develop a technology that helps us grow better nutrient food, so our bodies are taken care of longer, so we get more out of this life. And I really do think there's a big thing with technology and where we could go. If religion is false, then incorporating technology and trying to augment our bodies in a way to last long periods of time would be better than dying at any point and going into the blackness of space, the dark, going into the dark matter of the universe or whatever. When so I would say, like, that's part of why I think that these topics are so important, is we don't want to have people who have really good minds for this or that going towards religious stuff, when they should be going towards science, and also to enjoy life, to it's like having a good time. I've drinking beer before. I haven't drank for a while because I've never had an issue with it, but like I know some people do. I've been at parties where some people get a little out of hand. It can break up families and everything, which I'm very against. I think families are very important. And so, if society mends itself, I might have a beer or something to celebrate. But like I feel like those are like some things that someone could be every once it is fun to drink with your buddies and have a good time and all that stuff. Like that would be stuff that's that's part of what we would be able to be here to do as the ascended monkeys. That's one thing too I think should change. Darwin, don't go descent of man, call it ascent of man. Let's change our wording so we're improving and going up rather than down. So I
haven't heard that one, but that's pretty good.
So I think like that type of we don't want anyone to miss out on life. We don't want anyone to.
Yeah, and let me interject a little bit, because I think that that was one thing that you touched on in your debate, because that was the starting point with your intro, but also the way you finished off. You have I think a big part of your concern, which I can tell comes from a very genuine place, is that you're concerned that that the at the very least a non-biblical focused or non-Christian perspective, and potentially the evolution perspective, is one that is like amoral and it means that life is largely meaningless and could lead to, rampant destruction and decay of society, that kind of thing. Is that fair?
To an extent,
and
yeah.
I'm not saying, and just because I know part of maybe where your hesitation is coming from is that, like, I'm not trying to say that you think that I believe those things or that you,
yeah,
like,
no.
I'm
Yeah,
not
there
taking
are
it personally,
definitely good atheists,
Right.
agnostics out there, people who have moral code, ethics, have a way of determining right from wrong, and want to work together with people rather than take the one-man army approach. There are, like, I think Hitler was misrepresented as a Christian. I think the God that he kind of talked about was not the biblical God, but he was very big on something called self-derived human evolution, which evolutionarily, like, if you get rid of the gene pool, the weaker gene pools, you're accelerating evolution from like Hitler, evolutionarily speaking, Hitler was, to the science with that. Like, he was
not really.
Because evolution says that, like, the weaker gene pools will fade out, right? Because the stronger gene pools will stay around, will reproduce more.
Not necessarily. No, it isn't necessarily about strength either. And actually, I'm kind of glad you got onto this, because this, I think, is getting to what I was talking about today when we were going back and forth in YouTube comments a little bit about misconceptions that you have about evolution. It isn't about that, like, the strong dominates the weak. That's for sure. Like, evolution doesn't have to proceed that way.
Yeah, there can be symbiotic relationships.
Well, sure. And in fact, because one thing that you said in our debate is that you suggested two different scenarios. And one scenario was, well, if we put worms in an incubator and we change the humidity, we change the temperature, shouldn't we expect over time that will evolve into something else, like a fish or a bird or something like that? And then you also gave another scenario, which was, if I hold a worm above a table and I drop it, and I do this several times, shouldn't that worm eventually evolve armor or something, or wings?
Yes, armor, wings, something like that,
right? To protect itself.
Yeah.
Now, okay, let's take that second example, because I think it illustrates a severe misunderstanding in your part about how evolution proceeds. Do you believe that if you take a worm and you just drop it repeatedly, that we, as those of us who are proponents of evolution, that we would predict that there would be a response in that worm that would be like that worm should evolve some sort of novelty to deal with the fact that it's just falling over and over again?
I think it would evolve something that would allow it to be able to endure the hits better. Whatever it may be, whatever this population of worms, if you're just picking up thousands of them and dropping them, whichever ones are better, are able to embrace and don't get knocked out or die from it, the drop, they would stick around.
Right.
Okay.
Yes, that's getting closer, because the way that you described the experiment, it made me think that you believe that evolution happens in an individual. And it doesn't.
No. It's okay. Population.
It's on a population level. Okay, so at least we don't have that misunderstanding. So, right. So repeatedly dropping a worm isn't going to do anything, right?
No. That was I might have misspoken. But yeah, definitely. If you took a population of worms,
yes,
and drop them, certain ones would be able to endure the fall better,
right?
And then those genes.
Yes, and like I explained in our after show on Kaz's channel about the domesticated foxes. I don't know if you remember that story about the domesticated foxes in Siberia, that they were selecting for tameness in foxes. All they were looking for is just how tame the fox was. Relative.
They got like floppy ears, right, and a curly tail.
Exactly, but they didn't select for those things. They were only selecting for tameness, just how scared the fox was when a human approached. And they had hundreds and hundreds of foxes, and they assessed them all. They scored them. There's a range. Some were really, really scared. Most of them were kind of something in between, and then there were some who were like less scared. They didn't have quite the response that the average, or especially the really scaredy cats, did. So scaredy fox, I guess.
Scaredy dogs.
Yeah, scaredy
Canines.
dogs. Yeah.
Yeah.
So then they bred the ones that were being prone to being more tame together, and after 20 some generations, they had now a fox that was like basically tame, and acted essentially like a dog. In fact, I can bring up a video because it's kind of cool. I don't know if you've ever seen it, but for the audience, they might like this. But the thing is, like, over enough generations, with this kind of selection pressure, you can get this kind of change. Okay, yes.
Can I while you're pulling up that, because that would be definitely cool to see. If I could throw this your way, so this tameness of this tame population of foxes that got curly tails, floppy ears, could this have been, do you think it would be possible that if we took a different population of tame foxes that had different genetics, that maybe the expression of tail shape or ear shape would be different, and you would still keep the tameness?
So possibly. We don't understand all the underlying mechanisms of domestication syndrome, but I have a paper that is particularly interesting. I know Tecumseh Fitch was one of the authors on it. He's a professor at the University of Vienna, on the domestication syndrome and what he thinks is actually underlying. And I'll get to that punchline in a sec. So here is domesticated fox. And they're going to start with the non-tame ones.
Puppy dog.
Yeah.
Canines.
There's some noise here, so I'm going to unmute.
I love dogs. I'm a huge dog guy. Foxes are not dogs, but like I love dogs.
Mm-hmm. Not a happy fox.
Yeah, he looks like he'd rather not be in that cage.
I'll skip ahead. Here's another one. But then, so here's one that's very scared, right? So you open the cage and then it just wants to escape. And then here are the domesticated ones. And you can see the ears are sort of floppy.
Mm-hmm.
And then this one is like white in color.
Wow. Oh, he's happy too. His tail's wagging.
Exactly.
Short. It looks like a shorter tail too, though.
Yes, the tail is shorter as well, and they tend to be a little bit smaller too.
Aw.
Yeah, and there's one that wants to play. Yeah, so stop sharing here. That
very cool.
Yeah, after just 20 generations, they had that shift. Now, I'll have to look up. I'm going to look up if I can find this article from Fitch. Yes, here we are. Okay, so I have a video on this, actually, the domestication syndrome from a neuroscience perspective. But this is Richard Wrangham is a professor at Harvard. Adam Wilkins is at in South Africa. I believe the Humboldt University in Berlin. I guess so, maybe. And then Tecumseh Fitch, as I said, he's at the University of Vienna. So they have written this interesting article on the domestication syndrome, where basically, like with the domestication syndrome, you have a list of features that kind of go along with domesticated species. And this includes depigmentation. So such as like, especially like getting white patches. You saw that one that had like white skin, floppy ears. And here's all the different animals that have floppy ears, reduced ears, shorter muzzles and teeth, being docile. So of course that's true in all domesticated species. They generally will have a smaller brain or a diminished cranial capacity, which is where like part of my interest comes into it.
This is to speaking about like the caninus family.
Mm-hmm.
Okay.
Well, no, so dogs are in here, but as well are cattle, goat, sheep, pig, rabbits, gerbils, and when we say smaller, okay, we're referring the domesticated version to the version that they were bred from. So like with rats, like there are domesticated rats, we compare them to wild rats, they have smaller brains. Of course, they're going to be more docile. Some of them have smaller teeth, like domesticated mice and dogs and pigs. They have smaller teeth than wild mice, than wolves and like hogs.
Are you just curious? Have you heard a term regression to the mean?
Yeah.
Or like if you
this isn't about the mean. So I think what's happening here, and this is their explanation that they have, is that they argue, and I think that there's probably some good reason to believe that this is the case, that when you're selecting for tameness, you are selecting for a reduced sympathetic nervous system response, which is our fight or flight response, as well as a reduced adrenal system response, which produces cortisol and corticosterone, which are the stress hormones, right? And so
for emergency situations.
Yeah, exactly, right? Fight or flight, right? So it's got to be engaged. And so when you are selecting for tameness, there's variability in how robust the sympathetic ganglia are and how robust the adrenal glands are. And generally, these things are going to be similar because they actually arise from the same part of the embryo, which is known as the neural crest. So in the early embryo, when the nervous system is developing and it gets folded in, and then you get the neural tube, there's a top part of the neural tube, which is the neural crest. And you have these cells that migrate away from the neural tube, and they end up being other things, such as melanocytes, which are critical for pigmentation in the skin, odontoblasts, which are the things that give rise to the teeth, as well as chondrocytes, which make up the cartilage of the ears, as well as cartilage in the tail. And then, of course, like it's there could be effects that also impair the size of the brain as well. So what they argue is that when you are selecting for tameness, you are selecting for genes that would have ended up in compromised development of the sympathetic ganglia, as well as the adrenal glands. But you're also just getting these other things that hijack along it because they arrive from the same part of the embryo, such as the melanocytes. So therefore, now you get the pigmentation changes, and then the cartilage that makes up the tail and in the ears, as well as the odontoblasts.
They all kind of have an association with that crest cells, and these are things that in the wild you would need these to have an advantage over what it is that you're trying to catch like as prey. Like you need the teeth and the adrenaline, the ability to book it in a hurry.
Sure.
But if you're domesticated, you have people to help you get your food.
Exactly. So, right. Exactly. And so there's now, but this is the thing. Variation exists in a population. And human beings, of course, if they are selecting for tameness, they are selecting within that variation, and they're doing a much more robust selection procedure than, say, natural selection can do. Because natural selection also has a fair amount of chaos that's built into it. You might have an animal that's particularly well suited for a particular environment, but it got struck by lightning, and too bad, you don't survive. That has nothing to do with genes. It's just, too bad. Like, that's just how it is. Whereas with humans, especially with this Russian experiment, they were going through hundreds and hundreds of foxes, finding the few foxes that are particularly tame, and then breeding them together to then get the certain genetic mix that exists within that population, making that subpopulation now very distinct, and so that they now have these features. But they weren't even selecting for these features. That's the amazing thing about this. that's the thing about the domestication syndrome.
They were targeting the tameness, and then they got these kind of tag-along features to develop as well, alongside.
And this is evidence of just how pleiotropic the body is, that you don't select for a thing, that you could select for a thing, but a lot of other things can go along with it that you might not even necessarily be selecting for. The body, all animal bodies, they're not machines. There's not a single machine that works this way, where if you change one thing, all of a sudden you get all these other things that can change with it, and like the animal will be mostly fine. But like so much can change because you've influenced this one thing. And we see this at the molecular level, where there's so many different molecules that will interact, and you know, it's a mess whenever you see, and you may be seeing this in your organic chemistry class that you're talking about before we got live streaming, but you'll probably see these flow charts of like, well, this acts on this, and then this acts on this, and this acts on this. And yeah, sure, those things act on each other. But the thing that your professor isn't telling you about, because maybe they don't even know, is that there are lots of other things that influence and act on it as well. And it's a web. I hate these molecular mechanisms. I work on them, and I deal with it myself, but I hate them because they ignore the vast complexity and the web on the molecular level of, well, if I change this, so many other things change. Like they'll give you like
lines on a paper, for like reaction steps. You'll go through and it'll be like, you have this reaction, line down, arrow down to the next reaction. But yeah, it really is kind of more, it's like three-dimensional with outside aspects that are all involved. And
that's exactly. And the body works that way too. So a change in the sympathetic nervous system is going to result in other changes in other parts of the body. And especially because if you are inducing a change within, say, the sympathetic nervous system to make the animal less, aggressive and to make it more tame, well, the way we're doing that is that we're affecting something in development, such as the neural crest. And unfortunately, there are other consequences of that, because there's other things that come from neural crest, such as melanophores and melanocytes that make up pigmentation, or odontoblasts, or the chondrocytes that make up the ears and make up the tail, right?
Well, and looking at it from like a natural selection perspective of like survival and being able to survive, your ability to survive as any particular animal, is going to be hugely dependent on how well your sympathetic nervous system is how quickly it's able to fire, because if you're being chased, you have to get away or you don't contribute to the next generation. Yeah. I mean, if you haven't already had kids, had offspring, or in an animal's case, like so having such a tie to the nervous system, like, to me, that is so fascinating to see how like these key features, like these features that will help you survive better, are tied to the development of your nervous system, your sympathetic systems, to be able to say, hey, everything developed this way, and put all of these energies going out to all these things that are all working together to accomplish the same goal of survival, of living.
Well, yeah, exactly. Or that if you accept the tenet of that animals, there's variation that exists within populations. That variation is at least in part and largely dependent upon heritable characteristics such as genes, that some variations are better suited for particular environments than others. And that by better suited doesn't necessarily mean stronger, doesn't mean smarter necessarily. It's just by better suited, what we mean is that they can survive and that they can reproduce. And that reproduction part is the key part, that if that individual can reproduce, or that group of animals can reproduce quite a bit, well, that means more of their genes make their way into the next generation, which means that you get changes in allele frequency over time. And just over a short, you can see 20 generations, you can get in,
which is not long. 20 generations is pretty dang quick.
Exactly. And of course, like, again, as I said, artificial selection pressure is hyper selection, right? Like, it is as pure selection as you could possibly get for the most part.
That kind of
randomness.
Yeah, that lighter fox is going to have a more difficult time keeping itself warm. I've
Yeah,
with
maybe.
living with my roommate, I've finally lived with a cat, and for the first time ever, I really like cats now. As a consequence of that. But but
They,
like I'll see when she
there's
lays
the claim
down.
that cats are domesticated. I don't know if I believe them, but
Yeah,
uh,
she's definitely a feisty one. Now, when she
uh-huh.
wants to be, they're smart. Like
Yeah.
she knows how to like work you if when she wants to.
Mhm.
But she'll when she lays down, she'll like curl her tail around her and like bundle up and create her own heat. And like an animal like that fox with the shorter tail is gonna have less ability to, especially in like a cold climate like Siberia, it's gonna be really cold. And so having
Right.
those, you almost become, through artificial selection, it's almost so hyper that the creatures that you have that have these traits now are now they're dependent on the ones who gave them those traits, who breeded for those traits. Now, because if you let them go back into the wild, they will get outcompeted. They do have a
Right.
less responsive sympathetic system, and then lack the features to like the teeth and everything, and
Sure.
to
But
compete.
and then you could imagine that this robust selection that's occurred that has allowed for the domestication syndrome. Obviously, for human beings, it's fine. Like, we'll continue to work with these animals because they're tame. That's the thing that we want. And
Yeah,
the
we want to have them
coloration,
as
if
pets.
anything, it's kind of cute, and
Yeah.
people like that part. So if anything, that just makes those animals even more desirable. But let's say we didn't. Well, then you could probably shift the population one direction or another over time. And that's where, of course, you're going to get different breeds and different variations of these domesticated species. Like, there's so many different kinds of chickens, so many different kinds of dogs that obviously have been selected for in various ways to create this enormous variation.
Yeah, dogs
So,
can
right.
fit a whole slew of niches just
Sure.
based on what traits we're looking for. A
Mhm.
companion, or if you're a hunter. I I
And the
I'm
variability on the genetic level
it's
between the different dogs is incredibly low, very,
Yeah,
very low.
but the
Yet there's this
expression is
huge range.
enormous,
Hmm.
and we
Yeah.
can, yeah, if you want to breed for like for a bigger dog, you
Yep.
can do that, or a purse dog.
Sure.
Like the pliability of genes is
Mhm.
really immense. Like
Yeah,
they're
and there's the Dog Genome Project, which has done an enormous amount of work on figuring out precisely what genes are involved with certain kinds of behaviors. Although they are just starting to get to some of that stuff, but especially on morphologies, and also figuring out like which parts of the world the dogs came from. Just looking up the Dog Genome Project, it's a whole slew of really interesting work. There's a relatively famous geneticist named Elaine Ostrander who's done a lot of this work. I know she's not the only one. There are other people who have done this too, but her lab has done a lot of cool work. I mean, maybe I'm partial
she's
to her.
involved with this dog genome
Yeah,
project.
she's at the National Institutes of Health. She was at the University of Washington when I was at the University of Washington as a graduate student. So I'm particularly partial to
Hey,
her, but
can I
yeah.
jump real quick from there to
Mhm.
something?
Yeah.
What is it that got you interested in neuroscience
Oh,
and got
sure.
you wanting to do what you wanted to do? And
Mhm.
do you, well, like, what do you like about it? What do
That's
you,
great. It's a good question. So I came to neuroscience through evolution, actually. I was particularly passionate about evolution. I'll back up a little bit. One of the most important books that I read in my life, which changed my life a lot, was Cosmos by Carl Sagan. So when I was in high school, I was a big astronomy nerd. I really liked astronomy. When I was a little
nice.
kid, I wanted to be an astronaut. And then I was like, okay, well, I'm probably not going to be an astronaut, but I could do astronomy, and that would be great. So I loved astronomy, and I loved learning about space. The vastness of space was just something that was just so dumbfounding to me. And then Carl Sagan, I'm sure you know who Carl Sagan is, but for those who don't, right, he's one of these great science communicators of the 20th century. He unfortunately passed at too young an age due to cancer. But he's written some really, really great books. Pale Blue Dot is one of them. The Demon Haunted World is an excellent book, which I encourage anyone to read. But Cosmos really changed my life because I was reading all of his science books thinking I was reading about space, but he tricked me because Cosmos is about, and of course there's the Cosmos series that Neil deGrasse Tyson picked up on, but I read the
kind
book.
of inspired by Sagan.
Yes, right, exactly. It was because Sagan had made a series in the late 70s or early 80s or something, and I was too young to see that, but I read the book that was part of that, and there's a chapter in there about evolution and about natural selection. And I, you know, being a public school kid, I didn't learn about evolution, not at least in great detail that actually caught my attention. You know, this is a lie that often gets told, that like, oh, well, you go to public school, you learn about evolution. I would say that you don't, like, you don't learn about it the way you're supposed to, or that like really shares the amazing capacity that evolution has, and teaching the fundamentals of natural selection in a way. But he had such a way of explaining things that I was like, that's it. I want to go study this. So I became fascinated with the process of natural selection and biodiversity on Earth. So I started reading all about that. Stephen Jay Gould, E.O. Wilson, other people like that. And then when I got to college, I was like, okay, I mean, I'm definitely interested in the evolution thing. I also am pretty good at chemistry, so I'm gonna do biology and chemistry and see which one I want to figure out and do. And I ended up taking chemistry for chem majors my first year in college, and not liking it very much. And I was realizing like, nah, this isn't really my passion. So I'm going to study ecology, ethology, and evolution, which is the degree that was available at the University of Washington, University of Illinois at Champaign-Urbana, where I did my undergraduate. So, and in that, it's like ecology, ethology, and evolution. Those are three different things, right? You got ecology, which is study, you know, the environment and that kind of thing, population genetics, populations, those kinds of things. Ethology is the study of animal behavior, and then evolution, which is, of course, evolution. You had to learn about a little bit about each of them, but you could specialize in whatever you wanted. So I wanted to specialize in evolution, but it meant I had to learn about ethology and I had to learn about ecology. Well, I ended up taking an ethology class that really captured my attention. It was taught by a really great professor at University of Illinois named Gene Robinson. And I wanted to join his lab. So he studied animal behavior, studied the behavior of honeybees. And so I ended up joining his lab and working as an undergraduate research assistant in his lab for 2.5 years, learning about the behavior of honeybees. And I figured like, okay, I can marry these two things. Like evolution, I'm fascinated with evolution, but I don't necessarily, I also like animal behavior. And if you want to understand animal behavior from an evolutionary context, some of it has to involve now looking at the brain. And that you in order to understand the mechanisms of behavior, you got to get into the brain. And so that's what drew me into the brain. And so Gene, he was, you know, he is like, he's a member of the National Academy of Sciences, which is a very elite group of scientists. That you know, he was also the director of the neuroscience program. He was the head of the Genomics Institute at the University of Illinois. He's like a top scientist. You know, hundreds and hundreds of papers that he's published. You know, he's a pretty amazing guy too, and very nice too. Very generous with
excellent
like, support that he has to his students. But he
good to hear. Professors go a long way in inspiring students.
Made a huge difference in my life for sure. But but he studied honeybee behavior, like how do they act on a colony level, on an individual level, the fact that they do in-hive tasks when they're young, and what happens when they get older, and then they start to do out-of-hive tasks like foraging for nectar and pollen. Like, why do they do that? What happened? Why are they doing that at the colony level? Why are they doing that ethologically and for like an evolutionary perspective? But what's happening physiologically with them? And what's happening in their brains, and what's happening at the genetic level to control this as well. So he started the Honeybee Genome Project, and you know, was the head of that. So he was part of sequencing the honeybee genome. I was there when that was happening. I was not involved with that, but I did learning and memory experiments with honeybees. So we would do olfactory conditioning with honeybees, where we expose them to 2 different odors, and we give them either a punishment or a reward. And do you know how you punish a honeybee?
This is gonna be a funny joke. What's the punchline? What is
Yeah, you insult the queen, right? They say something nasty about the queen, right? Yeah.
Well,
your mother's so fat, you do a mama fat joke. Yeah. No. So you just
A
give it a
knock knock
little
joke.
Yeah. You give it a little salt solution. So they don't like salt, but the
Okay.
reward is sugar. And then but they learn, and they do this cool behavior called the proboscis extension reflex, where they stick their tongue out. They go bloop. If they have learned the odor is associated with a reward, they'll stick their tongue out because they're expecting the reward. So so that's what I was working on as an undergraduate. And then that got me into neuroscience, and then I started studying the neural circuits of singing behavior in songbirds. So that's what I did my PhD on. So songbirds, they actually learn their vocalizations, and this actually can kind of bring us to the vocalization and learning and memory, or the language thing that we were talking about earlier over YouTube chat. Like how we know like that emotion can be manifested in the brain. Talking to a neuroscience, obviously I can get into the details of like how that comes about. Hopefully that was
you definitely addressed that with
See, this is the thing, like a lot of people will say like, oh, I mean, how can you explain emotion in the brain? I'm like, well, we do know a lot about it. We don't know it all. I won't pretend to say like we've got every detail worked out, but there's an enormous amount that we know about emotion in the brain.
Which does, I mean, from when you do look at a word like happiness, it is gonna run like happiness, love, and peace, and these things. When you see them, it is gonna make you feel, and it like immediately you feel an emotion of those types of emotions that does activate your brain.
They draw up certain emotions in us that are pleasing or that make us feel good, and then you can think of other words such as like, you know, nasty or ugly, and that these are things that draw up negative emotions for us, and that we have an inherent salience that we denote to those words, right? Now, the cool thing about that is that you can do an interesting test, and I don't know if you're familiar with the implicit bias phenomenon. So there's this thing obviously about explicit bias, like someone can say, you know, this is awful to say, but like, oh, I don't like you know, I have certain things, negative things that I believe about Black people, for instance. And someone might spout off and say a lot of horrible things about Black people. That's explicit, right? You're saying those things. So you're clearly biased towards Black people. But most people know to not, at least not say those things. And most people genuinely don't believe those things.
Yeah, don't have any.
Most people
all
don't.
People are, people are people.
Yeah, you got old people and certain other people. But for most people, they don't. But the thing that's really interesting is that there have been tests to look for implicit bias. So the way they started with this is that, okay, you do a word association test. So basically we have words like, you know, peace and love and happiness. And then we have things like war and nasty and ugly. And all of us will know like these things are good and then other things are bad. Now, what they did is that most people, there's a psychologist, I forget her name, but she's at Harvard right now. But what she did is that she took a word list of words related to flowers, which most people have good things to think about, and then words related to bugs, which actually most people have negative feelings about. And then she gave them this word list, and every time a word flower came up, the subject was given a choice between like a good word like peace or a bad word like ugly, and then they were asked to like pick the good word. So when a flower word showed up and then they had to pick a good word, they did that. And then if a bug word showed up and they were told you got to pick a bad word for those, then you pick the bad word. And so then they test like how quickly they can do that. Great. Okay. It turns out that if you flip the task and you tell them, okay, for now, if every bug word that you pick, you have to associate it with a good word like peace and love. And then for every flower word that shows up, you have to pick a nasty word or an ugly word. Then it actually becomes much harder. And then so people take much longer time to do it. They make a lot more mistakes when they do it as well. So there's an implicit bias that most people have, that they think flowers are nice and pretty and lovely and bugs are nasty and gross. And when you force them to flip it, then it becomes very hard.
So your biases can actually get in the way. They can hinder you from being able to respond quick to something if needed. To be fair, though, to the other side, like having a fast, peaceful reaction or beneficial reaction towards, you know, treating others, it's good to have that be as quick as possible.
But then here's the twist. What they did is that now they gave these subjects a list of white people names and black people names, traditional black people names and traditional white people names. And what they find is that the majority of people end up doing an easy job when they have to associate white people names with good words, and they have and black people names with bad words. But when you flip it, they take a much longer period of time to do it. They make a lot more mistakes, and that's implicit bias. And these are people who genuinely believe, like, they would never say, like, you know, that I'm racist, right? Like, they might be NPR listeners, and you know, they've got tote bags and they recycle and all this stuff, and they might say they had black friends and all this stuff. I mean, I probably do, but it turns out most people have this implicit bias that's built in, and that's implicit bias.
Wow.
Yeah. So yeah, it's super fascinating stuff. There was a recent podcast episode on Hidden Brain, which is a great podcast. He does a great job. Shankar Vedantam is the host, and
Chris, you're smart. You're really smart, dude. You're like,
but this is the thing about the brain, right? Like, it has all these connections and patterns that get put in and are stuck there, and you don't even necessarily think about them, and they're still there. And we even know some of the underlying circuitry that is involved with that. Obviously, we don't have it all worked out.
It's like creatures of habit, right?
Yeah,
like,
right. That these patterns get built in, they get entrained, and
you're strengthening the neural pathways as well. Like you're deepening those connections, strengthening the neural connections,
right,
and activating them more, allowing more pathways to build to these access points where more and more events can trigger the same, like, funnel to one point in your mind where you have this memory or this response, this emotion, this feeling.
Right.
Neuroplasticity
and a lot of it happens
incredible.
Much earlier than you think, too. That's the other scary thing about it, is what their work shows, is that they thought that, like, if we look at little kids, they're going to be kind of open to accepting people of the other race, and it turns out that they're not. That even by like 6 years of age, they just kind of automatically gravitate towards choices that would put them towards, like, their own race. And this is at least amongst white kids, and that's at least the study that I'm familiar with.
Where were these white kids, quote unquote, were they living in predominantly white areas?
Yes.
Okay.
Yes, predominantly white, and actually quite affluent and very intelligent, and not like an area that's, like, you know, filled with the KKK or anything like that.
Yeah, just kind of like a okay, I got you. It wasn't like a diverse kind of
not particularly diverse. No, it's certainly not with a lot of black people.
Okay.
So this was Cambridge, Massachusetts, which was the study population, you know, which is where, like, Harvard is, and it's very ritzy and expensive, and there aren't a lot of African Americans that live there. Anyway, yeah. So where was I going with that? I did want to touch on one thing, because I know you don't want to talk for too long, which I don't blame you. It's pretty late here too, but I wanted to talk about the difference between abiogenesis and evolution, because that was something that you came up in the comments. And saying that you have to accept abiogenesis and then evolution. They're like two sides of the same coin, basically, right? Yeah, that's not. I'm putting words in your mouth.
Yeah, well, but essentially, yeah, their evolution is dependent on an abiogenesis event happening.
But it's not. You understand that, right? Like, you could imagine an alien came to Earth and seeded the planet with some single last common ancestor.
If that happened, then the alien would have had to come from somewhere. At some point, I mean, you could move it to another planet. I think you complicate the ability to try to show non-life to life happening. Sure. But if you do, say, maybe it's a silicone-based life form that wanted to make a carbon-based life form, and they put a seed here, and then we all pop up out of it.
There's an infinite regression problem there. I would agree. Like, eventually, at some point, you got to explain, like, the first life. Now, well, what if you just concede that it was a miracle, and that there, you know, it was
I believe in a lot of miracles right now being Bible based. So I do and that is so, like, I do, like, beliefs are for people to have, and people can believe what they want. And so like I don't have a problem with that, as long as people don't believe they can hurt others or anything like that. But I do think that if you are going to claim, because I think, yeah, you had said like we're talking about philosophy when we're talking about evolution versus creation, and I think that is truly the where these conversations belong is philosophy. I would say in like a religion class, religious discussion class or something like that. The difference that I think is that one of them is taught in science, especially at college level. It's kind of a gloss in public school, in like high school and everything. Sure. But evolution is taught as science. It's taught as fact. I've talked with professors who have told me evolution is a fact. Right. Biology professors who say they couldn't think of anything that would falsify evolution, which science is a requirement. But I think having a belief is one thing, but teaching science, it should be things that we don't have to infer, and especially to such an extent.
I mean, we infer everything.
I mean, both things are like something that is empirical is like if I drop my phone, it's going to fall to the ground every time. It's always going to go down under standard conditions, and so. So that would be one thing we can observe, we can repeat. That is not necessarily belief because we can see it, we can test it, we can watch it happen. But if you are going to say,
but the model that explains it, then like obviously that's where you diverge from most conventional science, right? We believe that there's like it's a differential that occurs within the space-time continuum, that masses are attracted to each other through gravitational force, essentially. And I'm speaking as
gravity wells and space-time fabric wells and gravity. I would say density.
Yes, and you talk about density differentials, but the thing is, like with evolution, and this is the thing that I think like why you can talk about the fact of evolution is there's so much overwhelming evidence for the pattern of nested hierarchies, that animals descended over time, that Earth is very, very old, that there are all kinds of transitional fossils that you can find, and that the mechanism is like completely plausible as a mechanism by which you can get extraordinary novelty that can occur over a relatively short period of time.
Yeah, and I do want to say like something like, and I don't know if this is a misconception that maybe some kind of more secular people have, but like a creationist worldview, at least mine, and like every creationist that I've talked to, like things like natural selection are not, we don't take that into the model. Like I think natural selection is an intelligent thing to have in species to allow diversity and survival in environments.
I mean, it's just a logical consequence. I mean, that's the thing that's
it's observed.
That's the extraordinary thing about Darwin's insight, is that it's almost like no duh. Like how else could it possibly work? I mean, it's just a series of premises and basic observations that obviously that's the case. Variation exists in a population. It is
absolutely. We see it. We look different from each other,
and that some but you look more like your parents than most other people, right? Because there's a shared genetic heritage. That was known for long before Darwin ever came on the scene, and that variation exists, and that of course, like environments, not everyone survives. Some survive, some don't. Those that have the variation,
humans can help each other survive. Just saying, we can help each other. But
like it's surprising, like how like simple it is. It's. I mean, I honestly think that's more like the fact of evolution. That is as close in biology as you can get to like a logical syllogism. That like, well, how else are you going to disprove that? I mean, I think that's also part of the reason why creationists minus bubblegum gun.
He apparently has kind of some very
polytheistic, goofy thing.
Yeah, he's
and if he's in here,
to each their own. I mean, to teach their own, but I am interested myself as well as I'm sure you are. Like, we want things that can explain things, help us better society and technology and improve in sciences and everything. That's what we want is models that can kind of help with doing these things. I was kind of curious what your take would be on, say abiogenesis was a miracle, and like the difference between dropping something and abiogenesis is we can drop something, see it fall, drop something, pick it up, drop it 100 times, it'll fall. But abiogenesis, like the claim of abiogenesis is non-life under natural conditions becoming alive.
Right.
And my understanding, like natural conditions to me is like if you have a beaker, you can mix things in it, and then you can change the temperature, the pressure, or anything like that. You can't like trap or extract or anything like that. You have to just keep using solution there. You can't have interference that coming from life, but like I know humans control conditions, so I don't say that as interference. Like if you boil water or something, that's still possible with like a geothermal vent. But with all of our scientists that are involved in origin of life theory, and I'll get to that in a sec, why I think it's important, because those people are very smart people. But we are not even really close to getting non-life to, under natural conditions, become alive, which is required. And if you say that this was like for people who accept evolution and a creator, if a creator had to start evolution and then all the empirical science that is supposed is claimed to support evolution, if that was all built on this foundation of a creator that started it, would that make evolution reliant on a belief system?
Like a faith statement, basically.
Yeah, like a creator kicked it off, and now we can observe the natural processes.
Not necessarily, no. Like not evolution anyway, right? Evolution still could proceed with the basic tenets of descent with modification via natural selection and random mutation from a last common ancestor. And even if that last common ancestor was poofed into existence by, a
creator of some
Vishnu,
sort.
Yes, or Buddha.
Yeah, and if the Bible was false, I mean, if the Bible was false, the Earth was a globe, evolution was true. If there was a creator to the universe, they don't seem to want anything to do with mankind for not giving us like a book or communication, putting us in a little speck of a speck of a speck around a speck of other specks and a speck of void. So I do want to say, like you had said, like natural selection and descent with modification, that is also something that like myself as a creationist, I ascribe to that, where you and I have a
creationists do. Yeah.
Yes. Yeah. Where we differ, just for clarity, where we are different is common ancestor versus common designer. Right. Which is where, but like a lot of, because there are smart creation scientists that have an understanding. There are guys who are creationists who say, when I go into the lab, I leave my beliefs out and I just study. What do I see? What do I observe? What am I testing? And I hope that everybody could admit there are some smart creationists out there, genuine creationists. There are smart atheists out there. And one of the things about origin of life scientists is that I personally have feelings about is like science is pretty difficult, especially like for a lot of people. And so having these people who are very smart and able to do these very complex things, studying origin of life, what these people should be doing is saying, here's the hypothesis. We can take crystals or we can take hydrothermal vents, test your hypothesis using natural conditions, simulate the conditions. If it fails, rework your hypothesis or discard it and go through every single idea for how life possibly could have happened. If we rule them all out and we're still not even close, life is so complex. One simple cell is just immense information, consciousness. There's so much to life that it's like,
I don't know.
If these scientists can't get close, wouldn't they be better off trying to develop a system to help? If you're working with molecules and assembling things, wouldn't you be better off trying to create a water filter that can get microplastics out of the water or anything like
people
that?
are working on that too, and I would say abiogenesis. It's not like there's that many labs focused on abiogenesis. There are some, and they're doing heroic work, and it's a challenging thing, and it's obviously something that's kind of well, it speaks to I know you don't believe that the other planets exist, but we're talking about life on other planets. And,
I can switch back and forth between. Okay. So yeah, I get what you're.
So, but the thing is, they are making enormous progress. In fact, Amiga Nuts points out that scientists are getting close, Pitch. In fact, there was a report that just came out this last couple of days ago. The Japanese Space Association or Foundation or whatever sent out a satellite that landed or took a sample from an asteroid, and they've analyzed it, and they found uracil, which is one of the key components of RNA on an asteroid flying in space, as well as a bunch of other like organic compounds. This was just published in, I think it was Nature this last week. And so astrobiology is doing work, but the people working here on Earth, like they're doing all kinds of experiments, and they get closer and closer, where like it seems like every single week they're coming out with a list of like the fundamental amino acids. Why, because there's more amino acids than the 20 that make up life. Why did life settle on the 20, and which are the ones that are the most fundamental?
Can we get them all in the right direction and to line up properly and that's enormous progress. Is there? But and then it's like, if you develop adenine, does the process that develops adenine simultaneously destroy any guanine that's in the mix? Like those are the things that would have to be, because if one destroys the other, then that hypothesis has to be reworked, because it wouldn't naturally work.
And I don't know those details. It's not what I work on, but I know that there's enormous progress being made
there. And I would say that from my understanding of it, there are, because I know like there was an article that got put out about like a synthetic cell, which was claimed to like, we're very close to getting life from non-life, but that was essentially taking an already made by life lipid membrane, and then taking a DNA coding, like DNA strands, and putting it into that membrane and calling it life. And that is, I think, like I think that's something within man's intelligence that we could do that type of a thing. I do think that one of the areas that I think is a big problem for abiogenesis is the would actually, I think, be the second law of thermodynamics, in that in nature we see things that lose energy, or that can gain order spontaneously. Like I know a snowflake is kind of spontaneous order. The thing about forming life is that you have to have both an increase in energy and an increase in order. And I do not believe, to the best of my knowledge, that there is an example where we can both increase energy and increase order. Increasing energy increases randomness, increases the destructiveness. So I think that is a huge hurdle just out of physical law for abiogenesis. And this is for someone who wants to, for any like atheist out there who wants to say evolution is strictly naturalistic processes. That is where an issue like abiogenesis being attached to evolution is required, because it's a continuum. Our universe formed, and then the earth, and then it was this primordial soup, and then life emerged. And so it is a continuum that needs natural explanation to be a naturalistic, viable worldview.
Okay. Yeah. So, oh yeah. Look, based also likes amyloid world. So I wanted to illustrate because there's so much emphasis on the RNA world hypothesis that RNA must have been like the original replicator, but this is a relatively recent review going over the amyloid world hypothesis. And yeah, so so there are people working on this, right? So the amyloid world hypothesis posits that in the pre-RNA era, information processing was based on catalytic amyloids. So the self-assembly of short peptides into beta sheet amyloid conformers leads to extraordinary structural stability and novel multifunctionality that cannot be achieved by the corresponding non-aggregated peptides. And so there's a bunch of work that's been done on this. There's actually still new work being done. And this is the basic idea of the amyloid sheets that you can
Chris,
get this like.
Would you be able to like these articles? I'll be rewatching this.
I'll put links in the description. I mean, because I'm sure you won't be the only one that will be interested in this. And I can send you the PDF for this too. But this is the basic model. Let me zoom out so we can see the whole thing. Right. So this is essentially the process, the model that this person is putting together. Right. And it's a model, right? But we do know that we can create these beta amyloids, that you can get these oligomers that can form. So you have peptide monomers, you get oligomerization, and then eventually the idea is that we can initiate an amyloid replication cycle that can lead to like changes that eventually can occur. And then eventually like this, so now this is like a heritable form where it can lead to like the environment influencing the amyloids that are developing, leading to the amyloid RNA protein cooperative networks. So like now we can have this. So instead of it being from RNA to protein, that it's protein to RNA. And so this can provide the stabilization because we know RNA and protein has a connection, right? That's the codons. And then eventually this can lead to compartmentalization with the membranes, right? So obviously like there's other work that this is just being summarized as a single box, and that eventually gets us to evolution. But this is like the basic process of the amyloid world hypothesis. So
I will definitely take a look at that because that is part of, and I guess I'm probably gonna have to split here just because I gotta work in the morning. But like
Sounds
this,
good.
To kind of relay this towards kind of maybe a closing. Thank you, by the way, Chris, for this discussion. I learned so much from you. You're really a fascinating guy. So
yeah, thanks. I like chatting with you. You're very genuine. You've got a lot of wrong ideas, but you come at it very genuinely. You don't like, I think a lot of creationists who have been around for a long time, they're not genuine. They know the arguments and they keep making the same mistakes over and over again. And you genuinely want to learn, which I think is great.
Well, thank you. And I'm definitely going to be checking out that freedom from religion critique.
The Easter challenge.
I'm going to check all of this stuff out. But like, I do consider that is substantial to anyone who wants to be genuine. I mean, if the Bible's true, we can't lie. So like lying, people do, but we're not supposed to.
How many animals were on the ark and, which are clean animals and which are not. Like largely that's kind of inconsequential to the Easter story. But like the Easter story itself is pretty important. And that particular article, I've always found to be like, okay, this is a big hurdle. And I'm going to definitely be deep diving into it. And it's something that he uses Easter because Easter, by the way, bunnies do not lay eggs. It's like with Ishtar and everything. So, but yeah, I'm gonna be looking into all this stuff definitely. And I did want to say too, like why Origin of Life. This is kind of back to like my schoolwork is I do want to write my capstone paper on the metabolic development of our metabolism's development throughout time, and if evolution is a science is the true way of how we got here and has the evidence for it and everything, or I mean even regardless it's what's taught in school, so things have to be through that lens kind of, at least while you're a student. But
I want to write about the development of our metabolism, starting from the beginning. What were the first things that life prioritized as the most important for metabolism, and then how did we develop our GI tract and everything, so we're able to absorb nutrients, extract things out of different foods, and then apply that into our cell health. And
well, yeah, I think, I mean, my understanding is that the basic fundamentals of molecular metabolism long preceded before there was any sort of GI tract or even a multicellular organism.
Yeah.
Right. So,
no, yeah, this would have to be done in the single cell.
Right.
We're
talking about
first
billions
life.
of years of single cell life, and
The
that's
Luca.
where all of it was worked out, which is tough then to sort of figure out exactly how all that evolves, since you're talking about molecular events and genetic changes that occurred billions of years ago where we have no fossil record. The only thing we can do is compare extant living organisms and, you know, try to piece together what occurred 2 or 3 billion years ago is a challenge without a doubt.
What are the most similar populations of creatures, organisms today that might have resembled that earliest life? Because even now, like they're saying, the simplest life to now, like the cyanobacteria, is probably far more complex than the proposed first life. I know there are certain like limits to how many genes you can take out and still have a functioning organism.
Right.
But I think that like what came first, metabolism or reproduction, or like I think there are some of those hurdles. But I want to thank you for this, really. And can I end it with like a you know, you ever heard like what came first, the chicken or the egg?
I have heard that. Yeah.
Okay, so I have a hypothesis to that.
Okay.
So I think that what came first, the chicken or the egg? It depends on if you're an evolutionist or a creationist. If you are a creationist, God made the chicken, the first chicken, and then it laid the egg. And if you are an evolutionist, egg-laying things were before the chicken.
Chickens, yeah.
So
it's true. I think we have a definitive answer depending upon worldview.
Yeah, right.
It's true.
So,
all right, good deal.
Thank you very much,
Good
Chris,
you,
and have a good night, man.
Yes. Yep. You too. Take care.
Likewise.
Right. Anyone in chat, if you want to pop in while I'm here, happy to have you join in. If someone wants to talk, there's only a few people watching at the moment. So that was good. Yeah, I mean, Nathan's a great guy. He's very, like, I do really mean that. I do think that he comes at this from a very genuine place, and a lot of creationists that I feel like that have very prominent perches are not. They are arguing out of bad faith, and they know that they're wrong, and they are doing it to argue a point, and they don't care about what is genuinely true, what is real. They don't care about seeking truth through like actual inquiring things. Oh yeah, let me send the link again. Link. Grayson, if you're going to join, that would be awesome. Hopefully that works. Maybe it won't. Hold on. Wow, I got a lot of tabs open. Should be it, I think. Oh yeah, there we are. Boom.
Hey, Dr. Chris.
Yo, how's it going?
Going well. I only caught the last few minutes of your conversation there, but it sounded very interesting.
Yeah, I mean, we were chatting. We had gotten on our modern day debate page. He had replied to a thread that I had replied to at one point, and then so I got a notification on my email that he was arguing with someone. So I jumped in, and that was today. And so then but I said, hey, why don't we just talk on stream? I haven't created enough content lately anyway, so I thought, like, instead of doing this over YouTube comments, which, like, kind of pointless, let's just make a, like, actually talk. And because we hadn't really touched base either since the debate that we had too, so there were a few things that I touched on. And Nathan is a really nice guy. He is.
So the only two things that I heard being brought up towards the end there were abiogenesis and the second law of thermodynamics.
That's right. Yeah. And okay, I'll be honest. I was reading something when he was talking about second law of thermodynamics. Do you want to add in something? Because there's your comment there.
Yeah. Well, I find that, in fact, about gravity interesting. Like, he was just sounded like he was just talking about like examples of things that, like, didn't obey the second law. So I brought up gravity as an example there, just because it's one of my favorites. I don't know if you're familiar, but one of my favorite physicists, Leonard Susskind.
I know Leonard Susskind.
So he's brought this up in one of his lectures. That's where I learned about this, which I always find a super interesting fact, that gravity doesn't really obey like the second law. It doesn't really have a very strong interaction with like with entropy, or like always fueling the increase in entropy. And the example that Leonard Susskind brings up is, like, basically if you had like a frozen star, right? So it's no longer doing nuclear fusion. It's not expending any energy. It's like frozen. But if you introduced like a planetary object to that, it could form, like, the gravity would form into a stable orbit, which would decrease the overall entropy of the system without any energy expenditure.
Sure.
So there's an example right there of the second law apparently being violated, because you have this closed system of these two celestial objects. There's no change in the total energy of the system. There's no, like, transfer of energy from one object to the other, but the total entropy has gone down.
Right.
So that was an example that Leonard Susskind brought up that I just find very interesting. Like, as a thought experiment.
I hadn't considered that. And it, in some ways it makes sense. Gravity is this really mysterious force in many ways, in that it doesn't necessarily involve energy transfer, but it obviously involves substantial effects on energy.
Yes.
And
and entropy.
Yes, and entropy. Yeah.
I think it's a little bit of like a side issue, because most of the time, like, the whole, like, evolution violates the second law claim can be shot down quite easily.
Pretty easily,
by just like, the earth is
not a closed system.
Yeah, exactly. Like, the second law only applies to closed systems, which somehow that message has not gotten through to the majority of creationists that bring up the argument.
They still bring it up. A lot of, if you ever bother to read, like, bad arguments that creationists shouldn't make, that will often be on that list.
Yeah.
That other creationists make, right? Like,
Well, I've also actually, funnily enough, I've had this argument actually the most intense with not a creationist, but like someone, I don't think they claim to be an atheist, but they are always in the comments and the live streams arguing with the creationists. And they're saying, like, no, you idiot, like, the second law only applies to the universe. And you can't use the second law to talk about any system that's not the total universe. And he claims to be a physicist in making these claims. So in that conversation, that's actually what caused me to do the most research into the whole relationship between the second law and evolution, because I had to show him actual papers from biologists and from physicists, most pointedly, describing the second law and, like, the changes in entropy for biological systems. So, like, just showing that you can calculate the entropy for any system, open or closed.
That's right.
Has a calculable entropy level and a change in entropy.
Right.
And so I found a very interesting article that was actually calculating the change in entropy as a result of biological processes per kilogram of human, like, tissue. And they were calculating that it actually changes over time. So the entropy change as you get older is actually less as a result of your metabolism than when you're like an infant or a child actually has the most drastic increase in entropy per kilogram.
Sure.
which was
I mean,
just
that makes sense too, right?
fascinating kind of results.
And that's the thing about the entropy as a concept can be applied in so many ways. The one way that I know of it is looking at the, so as a closed system, it to a certain respect, vocalizations of songbirds. So this is what I did my PhD on, where we were looking at how the brain circuits that regulate singing in songbirds. In my particular lab, we were interested in how there are seasonal changes that occur in these species, right? Sometimes in the breeding, like when they're in breeding season during the spring, they're singing a lot. This brain circuit that regulates singing in songbirds actually gets bigger, and then in the fall it becomes smaller. And we were looking at the hormones, how they regulate this process, like changes in testosterone and its metabolites. Now, the main thing is like birds learn to sing. That's a cool thing about songbirds. So they use this circuit to actually learn their songs, and you can measure the entropy of an individual bird's song repertoire by just measuring the actual attributes of the song. And so as a bird it starts to learn its song, it tends to be quite variable, and we refer to that as plastic song. Its entropy is actually really, really high. It has enormously high entropy. And then as it gravitates towards like a specific song, it gets better and better every single time it sings that song. And so now like, the entropy on a given day, like a month later, is going to be less. And we actually we're literally like using measurements of entropy for that. And it's within an individual bird. And of course, like you'll have a number that you can present for that bird and across multiple birds to create an average. And we can report like average entropy changes in a population as they learn their songs. So you're talking specifically about the behavior that they're producing, which is pretty cool.
Yeah, that's really fascinating.
Yeah, I mean, right. So, of
maybe
course,
you're the person to ask this, but if you have some knowledge of songbirds, but I had heard that one of the proteins that's involved in giving songbirds the ability to make their songs, I think it's involved specifically with like their tongue's ability to formulate the different, like, I guess, syllables of the song or something. But I had heard that one of the proteins that it gives them capability to do this was independently evolved in birds and in the human lineage, and that humans have the same protein that we use to like for our tongues to get the ability to form words.
So I think I know what you're talking about.
Okay.
So you might be talking about FOXP2. Is that right?
I remembered it started with an F, so that's a good sign.
So I actually worked on this. I have a paper on FOXP2 and songbirds. I can bring it up, actually. You're going to give me an opportunity to brag about myself a little bit, which is...
Well, I heard that it was evolved independently in birds and humans, but it's roughly the same protein.
The birds do not have the same exact amino acid sequence as humans. That is not true. It turns out that like humans, the human version of the FOXP2 has undergone some positive selection as best as we can tell. It's a gene that does not vary an awful lot across mammals and across all vertebrates. It's a pretty important gene. It's really important for aspects of brain development, but other parts of our body too use FOXP2 as a regulator. It's the
same. Sorry, just to make sure that this is the same one that I was thinking of.
Yeah, maybe.
I remember that there was like a story of like a certain family, I think like in Pakistan, or Afghanistan, somewhere in the Middle East. What?
Scotland.
Okay, well, I remember that there was a family that did not have this gene, and like they are like technically able to speak, but like they cannot, even though their vocal cords work and everything, because like their brains there's like a disconnect between their brains being able to inform their tongue for how to form the words.
Yes, that's essentially right. And it is a British family. It's known as the KE family that has a mutation in their FOXP2. It's a truncated version of FOXP2. So they have.
Do you know the ethnicity of this family? Maybe
I believe they're white
in Scotland, but maybe I'm just completely misremembering that fact.
No, I'm pretty sure that they're white. I'm pretty sure that they're like English natives.
Yeah, I don't know where I got Pakistani then.
Well, I mean, there's another mutation in that it might be. I'm not as familiar with like the underlying versions of, because I know there are other at least mutants of FOXP2 that have emerged. Simon Fisher is a professor at, I think he's at Oxford. He might be someplace else, but he's the guy who's done the most work on this, on FOXP2 in humans. We were looking at this in songbirds. So yeah, look, there's me right there. So this is a paper I published when I was in Germany. So we were looking at FOXP2 immunoreactivity in zebra finches. So I haven't looked at this paper in a long time. But basically what we were showing, I can skip ahead to here. So this is a cross-section of the area of the brain that has high expression of FOXP2. So this is FOXP2 immunohistochemistry. That's what this refers to. That's what that is. So we did immunostaining for FOXP2. This is the basal ganglia of the songbird brain. These are coronal sections. This is an adjacent section stained with crescent violet, which allows us to see like the cytoplasm of all the cells. So you can see like there's a lot of cells up here. These would be more, we refer to this as pallium, which is similar to the cortex in mammals. But the striatum is basically the striatum in both mammals and birds. And it's loaded. You can see that like, all right, there's a lot of cells here, but they don't really have a whole lot of FOXP2. But this area has a ton of FOXP2, right? And this, I don't know if you can see this, but there's this like lamb chop shaped thing here. This is an area of the song control system called area X. It's part of the striatum, which is critical for procedural memory. And it's necessary in order for birds to learn song. So PhD here, this is not that they graduated. This is post-hatch day. So post-hatch day 35 and post-hatch day 50. So these were birds that were 35 days old and 50 days old. And what we were looking at here is just what does the immunostaining look like if we zoom in and look at, the numbers, like the cells that have FOXP2 in them. So when we look really, really close, so this is a really zoomed in version of like looking in. So basically we're getting closer and closer, right? So this is zoomed out. This is this little box right here. This is the border of area X.
Yeah.
And so this is the inside and the outside. You can see that there's like tons of cells. And when we look really, really closely, we can see some cells that are really, really dark and then other cells that are light. So the arrow heads point to the light ones and then the arrows point to the dark ones. This is true inside and outside of area X. It doesn't really seem to change much whether you look inside or out. So this is in juveniles, okay? So what we're looking at here are not really the cells, but the nuclei. So FOXP2 is a transcription factor, and it affects the expression of all kinds of different genes. Now, when we look at adults, so these are now images taken from adults, one thing that we can see about FOXP2 is that it actually looks quite a bit different in adults than it does in juveniles. So in adults, here in a bird that was singing, now we see, instead of like us being able to circle area X because there's so many dark cells in there, instead we see there are basically no dark cells at all. In fact, it's almost completely absent. This is true, like there's very few dark cells even in non-singing birds, but one thing we do see in the non-singing birds is that there's still quite a few cells that are FOXP2 positive, but they're less intense. So they're relatively weakly stained. There's only a handful of really intensely darkly stained cells. So basically, one thing we can see here is that there's a singing related. So these birds were singing for 2 hours and these birds were not. And these birds that were singing, there's a down regulation of FOXP2 that occurred. So FOXP2 decreased the amount of protein that was there in these cells so that there are fewer of them. Like, so this is inside versus outside. You can see that difference. But if they don't sing, there's still some FOXP2 available. Basically, this whole paper, and I can skip to the end because I have a nice little graphic that I made for it. So this whole paper can be described in this model here. So basically what we were able to show is that there's a lot of FOXP2 positive cells inside area X, but there's a lot of them that are very intensely stained. And what we showed in this paper is that the cells that are intensely stained for FOXP2 are relatively young FOXP2 positive cells. So this circuit is developing and it's growing. And there's a proliferative zone where these cells are born and then they migrate into area X. And during that time, some of those cells, while they're young, they're going to have an intense nucleus for FOXP2 immunoreactivity, because FOXP2 is a transcription factor that promotes dendritogenesis. It promotes the dendrites to grow and develop. And it also promotes like synaptic development. And so as they differentiate and turn into neurons, you're going to need a lot of FOXP2 so that they can grow and incorporate into neural circuits. Once they get into a neural circuit, you no longer necessarily have to have them to have this really strong, intense FOXP2 immunostaining. But then we do see a singing-related regulation of FOXP2 expression. So it's there a little bit to titrate some of the change that can occur at the synaptic level that we do know that singing can induce. And so that's how FOXP2 changes in these songbirds. Now, to relate this back to humans, basically humans have the same kind of expression. Okay, so they also will have a lot of FOXP2 expressed in the striatum neurons, which is also critical for procedural memories, including vocalization. So it turns out that these circuits are basically the same. Birds and humans both have striatal circuits that are critical for learning procedural memories, such as vocal learning. It's just that this is convergent evolution, because obviously, like, it wasn't like a last common ancestor between birds and humans was also a vocal learner, because then you would expect a lot more animals to be vocal learners. Vocal learning is actually relatively rare in the animal kingdom, and it's only derived a few times. And, amongst mammals, I mean, it's only a handful of animals that actually have learned their vocalizations. So this family, they have a mutation in the FOXP2 gene, the KE family, and they cannot, and actually I've got to talk, could come up with it, but they cannot, they have what's called aphasia. So they struggle with speech production and not so much the cognitive understanding of language. So they can understand language pretty well, but the production of language is really, really compromised in these individuals.
Okay, so the convergent evolution between like bird lineages, like songbird lineages and human lineages is not so much in like the like enzyme itself. It's like, because from what I understood from what you said, like the actual like enzyme family of FOXP2 is shared between like pretty much all mammals, and like bird ancestors would
Right.
share the general enzyme class of like FOXP2, but
Right.
the actual like function within vocal learning or vocal expression would be like what was convergently evolved.
Yes.
Okay.
Right. And then it's ultimately you would be shocked to learn how similar all vertebrate brains are. I mean, it's maybe not you personally. You actually might not be all that shocked, but I think a lot of people would be. You think about brains being so complex that obviously, like, human brains got to be so much more complex than like a bird brain. And we refer to bird brains like
Yeah,
as a bird
I was about to
right?
say.
Exactly, right? But that's not the case. You'd be surprised at how similar it is.
So
So
question then,
Mhm.
so what you were showing with like the difference in the FOXP2 levels, like
Mhm.
before like a non-singing and then like after 2 hours of singing,
Yeah,
would there be a human equivalent to that? Like maybe like after 2
probably.
hours of like intense oration or
Mhm.
something, giving a presentation or having a debate or something,
Right.
maybe after 2 hours of that, would I
Potentially.
be depleting my levels of FOXP2?
So the thing is that not maybe E- actually I take that back. I think so. Um Especially if it's something where um it involved dopamine, which is something that's core to the way the striatum works.
Yeah,
So
you know
this
what?
is
I'll
a
just admit it. I get dopamine while I'm debating.
of course, so do I. It's part of the reason why I do it. Well, us debate bros are kind of addicts, actually.
Yeah.
Right? Like I'm doing this tonight because I need a hit. Like I was arguing with people in your video. Like, come
Yeah.
on, debate me, bro. Like, come on, come at me.
Yeah, my dopaminergic system needs a hit.
Exactly. So this is actually a slide I'd love to show. So this is the zebra finch here. They're very cute little birds. And what they're doing here is they're measuring levels of dopamine in the area X. So this specific song system circuit. And actually, maybe I should go back to the song system. So this is a figure I made up illustrating the song control system. So it's a series of discrete brain nuclei that regulate singing behavior. So this is anterior, this is dorsal. This is that pallium that I mentioned, which is basically like cortex. This is the cerebellum back here. This is the brainstem. And of course, we have other structures here. So area X is part of this. It's basically like a basal ganglia structure. The neurons we were talking about that are FOXP2 positive, they're striatal-like. They're medium spiny neurons. And this red circuit is known as the anterior forebrain pathway, and it's critical for the learning of song. And that when you mess this circuit up when birds are learning, you completely mess up their ability to learn song. But if you mess it up once they're adults, there actually are very few changes that occur in the singing production. But the descending motor pathway, like this is basically like premotor, this is like a motor-like cortical area, and then these are actual motor neurons that synapse on the muscles of the syrinx that produce the sounds in songbirds. This is a very much like a motor pathway, and if you damage these brain areas, then the bird can no longer sing, whether they're adults or juveniles. So this is yellow pathway. This is the ventral tegmental area and the substantia nigra pars compacta. These are the dopaminergic areas of the brain that synapse into the striatal circuits here. So these are the dopamine neurons. And for Parkinson's patients, they will lose these dopamine neurons, and then they can no longer like have a normal functioning basal ganglia, and then they end up freezing. And that's like one reason why Parkinson's occurs. So the amazing thing about this is like the circuit in mammals and in birds actually is very, very similar. The only difference is that in mammals, we have these two distinct areas of the basal ganglia called the striatum and the globus pallidus. And for a long time, like we knew that like there's this projection from HVC to area X and then to the thalamus. And we were like, okay, well, how can this really be the same as it is in mammals? Because they have two separate areas. It turns out in birds that basically they don't segregate these neurons from these neurons. They just mix them all up together in area X. And so they all get mixed up. So yeah. So the cool thing about this is that like you need dopamine to regulate this and to drive this. So you can put a bird, in a cage where you can measure the amount of dopamine that's happening in their brains in real time. So that's basically what this is showing. So there's a bird in here, there's an outline, and then you can measure like the amount of dopamine that's being produced like on basically a second by second basis. And then you can put a female in front of the cage, and then that bird will start to sing. And if he is singing towards a female, there'll be an increase in the amount of dopamine that's being released in area X while that bird is singing to the female. But if he's singing just by himself, so undirected song, there's not nearly as much of an increase. So this is another thing that we looked at in this paper, where we found that there was a difference as well as in like how much FOXP2 changes, whether they're singing directed versus undirected. And that dopamine probably seems to be one of the key parts to driving that change in FOXP2 expression.
Yeah, I guess if you have a visual on your potential reward,
right,
maybe that's gonna
so
the
that's
dopamine.
The thing about songbirds, especially. They're singing not because like they're debating with their debate bros.
They are laid.
Yes, exactly. It's one of the 4 F's that they're most concerned with.
So if you could like, instead of just espousing your pickup lines to the void, if you got a direct visual on your target, right. I could imagine that your dopamine systems are probably going to be elevated.
Exactly.
Wow, that's really interesting.
One last thing I'll say about the FOXP2, so one thing that you can do is that you can do genetic manipulations of like how much FOXP2 is expressed using what's called short hairpin RNA. So these are RNA constructs that will like interfere with the way this is produced. So like you can inject a lentivirus into the brain that will express short hairpin RNA. That'll block the production of these mRNAs so that you won't get any protein. And if you do this in a bird that is young, while the bird is learning their song, and if you inject these lentiviruses to knock down expression of the FOXP2, then they can still sing. So this is the short hairpin FOXP2, but this is similarity to the tutor song. So like the song that they were supposed to copy. And you can see that its similarity score is only 60%, whereas like the animals that were injected with a control virus, they have like a normal similarity score, which is about 90%. So interfering with FOXP2 expression specifically in area X, like during the phase when they would be learning, specifically blocks their ability to like learn song. So that was an experiment that someone else did.
And so do we know if that was like mediated by like dopamine levels basically being impacted?
This in particular, no. But the fact that dopamine plays such an important role in shaping the neural circuits of the basal ganglia, such as area X, certainly is likely shaping the how much FOXP2 is being expressed, and essentially like establishing which synapses should be preserved and which synapses should not. And that's the thing that is going to be the signal telling the brain, like, yes, you want to have this song working, and you want to match the song. So basically, the way dopamine works is that when a bird is singing, and if they're practicing the song and they sing it pretty well, and they're matching the template of the tutor, that feels good. It's like, oh, I did a pretty good job. There'll be a little bit of dopamine that's released that will act on the synapses in the area X, preserving those synapses to say, you know what, you did a good job at that point. We should preserve those synapses. And then if they screw up, there's going to be less dopamine that's released. And then those synapses that fired to make that song, that's an error signal. It'll say, you know what, maybe we don't want those synapses to be a part of the production of this song because you screwed up. So let's not do that. And that is the trial and error process that occurs for procedural memories. And that FOXP2 is likely one of those key genes that's involved in making sure neurons are plastic enough that they can continue to add synapses or subtract synapses.
Yeah, that's fascinating. And kind of review, I haven't so back in college I did a little bit of study of like the dopaminergic system, because I was actually working in a neuroscience lab where we were studying the effects of iron deficiency on like cognitive functioning. So we looked at specifically how like iron deficiency negatively impacts the dopamine system, but like dopamine metabolism production, signaling, and basically showing that like through its effects on dopamine, iron deficiency, like among college students, actually can mean the difference between an entire like grade point. So like an A or a B over the course of like a 4-year college degree. So like there's actually like, you know, it affects your ability to pay attention in class, to remember the information that you're learning, to recall the information that you have learned. It kind of impacts academic performance in every step, just based on like a nutrient deficiency.
Wow.
So it's pretty interesting stuff.
Yeah.
It's
I didn't realize
relevant for college campuses because the average college campus in the US among like college age women, the college age women of most colleges in the US are around the same level of iron deficiency as the average person in sub-Saharan Africa.
Wow.
So it's like a third world level of
iron
nutrient deficiencies.
Wow.
Mainly because
crazy.
of lifestyle, especially like, you know, like menstruating women are already at sufficient risk of iron deficiency, and you combine that with over drinking, bad diets, bad sleep schedules. It's just a recipe for anemia.
Yeah. Oh, that's super interesting. That's crazy. I didn't realize the incidence is so high. You know, iron is so key to so many metabolic pathways. It's not surprising to find out that it would be important for dopamine production. Dopamine is. Yeah.
And what you had said, I'm not that surprised to learn about the functioning of bird brains being similar to other vertebrates, because I was actually, for an upcoming video, I was looking at like the evolution of dreaming. And actually stumbled across a study that was looking at REM sleep in fruit flies. So they were noticing that there were analogs to REM sleep, because I'm honestly not sure how you would even measure REM sleep for a fruit fly because it doesn't have pupils, but apparently they were able to ascertain some sort of rapid eye movement or analogous behavior. So some kind of indicator that maybe flies are dreaming to whatever degree that would make sense for a fly to do.
All right, I gotta show you this. Then you're gonna really appreciate this. And you might want. Have you made the video yet, or?
Oh no, not yet.
Okay, all right.
No, just some like preliminary research because I find it's an interesting topic.
Right. So, okay. One thing we can do, another cool part about the song control system, is that we can look in these specific parts of the brain and measure actual brain activity.
Yes.
So you can use like these microelectrode arrays or single electrodes where you can like lower it into the brain, and these devices have gotten very sophisticated. So this is an example of a device where there's like a motor drive that's attached to a pipette, so you can record from individual neurons. But because there's a motor drive, you can change the position of the pipette in a live awake animal and record from different neurons. So you can record from multiple neurons at different times. So one thing about like change in activity for most brain areas is there isn't like a neural code. It's not like the actual pattern of the neurons is terribly important in most brain areas. So generally what will happen is, so this is what we call a raster plot. Okay. So this is like number of trials. Like we can measure, like say we put a mouse in a cage and we turn on the light and we're measuring their visual cortex. This line would be like when the light turned on, and you might see that like, so each little line represents an action potential that was firing.
Okay.
And obviously the rate of action potential goes up as the light goes on.
Yeah,
right?
Makes sense.
Yes, but the only pattern you can see is that at time 0 across the 20 trials that the overall rate increased. And that makes sense. And that's how a lot of neural circuits seem to work. Now the song control system seems to be particularly unique. So this is an example of a sonogram and a zebrafinch. This is like a visual representation of what the song is supposed to look like. So when they sing, we can kind of draw out what it looks like. This is, like frequency on the y-axis, time is on the x-axis, and like strength of the signal, the amplitude, is like denoted by like this color. Okay. So their song is actually not all that interesting, but it's stereotyped at least. So they basically sing the same song over and over again. And it's very, very similar. So the cool thing about that then is that you have this very, very stereotyped behavior. And if we're measuring in, say, nucleus HVC, and if we measure like from one neuron, so we're measuring from a single neuron and we measure it over and over and over again, that neuron is basically firing a few times within like about a 5 millisecond window. And then that's it for like basically an entire song. So it fires very, very precisely. And then if you measure from a different neuron, so some other HVC neuron, so you lower you turn on the micro drive and you measure from a different neuron, now that neuron fires at a different place. And remember, HVC, this is a brain area that's filled with hundreds of thousands of neurons. Even in a songbird, it has that many neurons in one HVC. So it's going to be the constellation of the activity of all those neurons working together that's allowing for the drive eventually getting to that vocal organ, the syrinx, that allows it to produce. Okay, so that's cool in itself that we have this specific code. And there aren't really good examples in any other animal behavior system where you find this level of precision related to a specific behavior in like a neuron that will fire at a very specific time.
Well, now I have a question about that, if I can.
Sure.
Because I want to ask about the infamous Jennifer Aniston neuron. Have you heard about that?
Sure.
Okay, so how does that relate to something? Because I've been told that there's a specific neuron in the brain, basically, that will light up whenever you're thinking of or talking about Jennifer Aniston.
Okay, well, first of all, I can guarantee you I have more than one Jennifer Aniston neuron. But, I've, especially because of my age, but not just that, though. No, so yes, Jennifer Aniston neuron or the Halle Berry neuron, and I'll come to that. So we'll get back to that, but let's touch on sleep first. So this is what RA looks like. RA neurons. This is the just to go back to the circuit. HVC projects to RA. These neurons are firing. They have like the single firing that occurs, and now it's being summed in RA. So RA is collecting this information from HVC, and now this is what RA looks like. So now any individual neuron seems to fire more than once, but they seem to fire at the same exact place across multiple songs, right? So this is a raster plot for neuron 1, a raster plot for neuron 2. And so now you can imagine you can create this like computational model of how this gets generated. All right. Now let me get to sleep. So I'm going to bring this back to sleep. So the thing about sleep, let me turn this off and bring up a different one.
Do you think that they'd ever be able to predict like what a sound the song would sound like based purely on which neurons were lighting up in which order?
I think so, yes. I do think eventually we'll get to that point.
Like I wouldn't think that would be so simple as like a correlation between this neuron means that this note is being hit. But
right.
I would think that you'd be able to like decipher that code.
Right. Yes, I think so. Yes. And that, ultimately this is going to lead to us making neurocomputing machines that will, allow us to do advanced processing of information in ways that will be very different than like the way a computer actually works, where it's ones and zeros. Instead, it'll be, action potential or not, but you'll also allow for like the computation that is multiplicity and exponential than what a transistor can do. That's some basic thoughts that I have. All right, sleep. All right, so this is now we're looking at the same thing, right? Songbirds, and we're doing recordings in songbirds. This is recordings from one songbird where we're measuring like activity in, I think that this is RA, no, HVC. So there's a neuron here in HVC that was firing a bunch, right? When the bird is singing. Now what they did, of course, you've got this electrode in the brain, and the bird has to sleep, right? So you let it sleep, light goes off, the bird eventually sleeps. If you continue to record while the bird is asleep, you will have spontaneous activity that matches more or less the same activity that they have when they're singing. So this is another example here. You can see the same basic firing pattern is there as well. So it's almost as if they're dreaming about singing.
Yeah. That's really interesting.
It's the same exact code. So if you want to look up more of this, Dan Margoliash, he's at the University of Chicago. I'm sure he's still there, but he's done a lot of work on sleep and the song control system.
Well, I know one of the most common hypotheses for why we dream is kind of like a simulation or a rehearsal for different events that could happen in our lives, or like, to sort of, go through information that you learned and sort it into usable categories, but also to rehearse for potential scenarios and how you would react, and kind of train some of your responses to certain stimuli.
Right.
But either way, it seems like there is some kind of advantage to dreaming over not dreaming. That seems obvious.
To consolidate. So it obviously allows for repair and restoration of our organ systems, but it seems to play a crucial role in memory.
Well, that's sleep in general, right?
Yes.
I find it like, because, if I've often thought about this, like if I was some kind of alien, right? Or if I was talking with an alien and I was kind of explaining to them everything that I know about life and Earth, and I'm just kind of going through, what our biology and everything. And then if I would just think that if I get to the part about dreaming, and every single night we become inactive, we lose our consciousness for a few hours, and then our brains construct a new reality that can be similar or totally dissimilar to reality, in which we all of our sensations feel real. It's a simulated universe that takes place every night in our dreams. I feel like the alien would just look at me like I'm crazy. Like that's some sort of superpower. Like, how is your brain constructing a universal simulator every night?
Universal simulator.
Like, it would sound ridiculous if
you might have superpowers. Yeah.
Yeah.
I know. No, it's and it's no wonder that like sleep has featured so importantly in development of culture, and dreaming is so critical to so much of like culture and aspects of religion and mythology, because it is essentially like an alternate world where we might be the hero or the villain, or fantastical things can occur, and it seems so real.
I do wonder, you mentioned that it might be possible at some point in the future to reconstruct a bird song based on just
the activity.
neural activity. So I wonder if one day we'll be able to, watch our dreams back on a TV screen, based on our neural activity from when we were asleep. So that would be a whole new level of fodder for therapists everywhere, I'm sure.
This is kind of related to that. So I don't know if you're familiar with what place cells are, but these are neurons that are found in the hippocampus that specifically fire when an animal is located in a particular place along a track that they know, like a maze.
Interesting.
So as an animal is going along the track, like individual neurons may start to fire when they're in that particular place. And so basically the idea is, as an animal will go along a track, like some neurons are going to respond and fire in certain ways at that place. So this is an illustration of what this looks like. I don't know if you can hear that. Can you hear it?
No, I can't. I don't.
Okay, that's fine. It doesn't matter. But basically, this is a mouse that's going along a track, and the yellow dots are different neurons. And so you can see, like, as the mouse goes along the track, now a different set of neurons are starting to fire. So now you have like a pink neuron that's firing, where there was blue over here, and there's and otherwise. So one interesting thing, though, I'm going to rewind it just a little bit. So as it goes along, and it gets to the end of the track, and I guess there's probably a piece of cheese or something at the end of the track, right? So once it gets there, watch what happens here. Did you see that? It's going to do it again.
You like kind of like where multiple neurons all light up at once?
All light up at once. And this is what's known as a sharp wave ripple. So it turns out that when a mouse is entered into a track, it will often do what's called a before sharp wave ripple, where it does the in a very, very short period of time, all of the neurons that are going to fire as the mouse is going to go along the track, they all fire in a very short period of time in order. And then when the mouse finishes the track, they do the sharp wave ripple in reverse order once they get through. Yes, and it all happens super fast, as you saw. Like, this is a sharp wave ripple in reverse order, within like 50 milliseconds. But it took like a few seconds for it to go through the whole track. Now, the crazy thing is that when mice are asleep, and if they've been undergoing these tracks and then been doing this training, like if they're going through a maze, we can track like how these neurons are firing. But when they're asleep, they do the same thing. And we can measure that. Now it's kind of sped up, which is also kind of crazy. So they've changed the scale here. Like, so the scale here is 1 second versus 0.2 seconds. So things seem sped up, almost like that they're sharp wave ripples that are occurring very quickly while they're asleep. Whereas when they're running, like, this is like the normal pace that the neuron is firing. So we can do this in the hippocampus, and we can also see similar kinds of activity in the visual cortex. So, different parts of the visual cortex in a mouse, when a mouse is navigating a map, different neurons in the visual cortex are going to respond differently to different areas because they're seeing different things. When we can see the same kind of basic pattern when they're asleep. So this is pretty good indication that they're dreaming about the map.
But again, it looks like the time interval is sped up.
And the time interval is sped up, yes.
Which is weird to me because I had heard that the like the perception of time, like the flow of time in dreams, was the same as in waking life, unlike in the movie Inception.
I won't argue that. I don't know. That could be. I'm not a sleep expert, and so I don't actually know. And I'm not entirely sure why it seems sped up here. Like there
well, I know the movie Inception, like,
yes, and Inception is like slowed down, right?
Yeah, but I had heard that it was
that it's the same.
Yeah.
I think it may be in fact sort of sped up to some degree. At least according to that's what that would suggest. And of course, this is in rodents. I don't know about data in humans. Now, the other thing I was going to say about this is, if you interfere with this activity in these brain areas, like particularly in the hippocampus, or if you interfere with this activity.
Wait, let me guess. It affects their ability to remember the direction to get to the food, like their memories
the next day.
Yeah, exactly. That would be my guess. So
that's it. So, and the same is true for songbirds. If you mess up their ability to do that spontaneous singing when they are asleep, and you're just kind of like activating the HVC during that time, that you slow down their ability to sing or to learn the song.
That's really cool.
So it's really important for that too.
Well, the last thing that I wanted to ask you about, just because you're the only person I've ever seen bring up the amyloid world hypothesis.
Oh, really?
And it's got to be one of my favorite hypotheses around abiogenesis, and I never see anybody bring it up. I feel like I'm like the only amyloid world guy out there. So when I saw you bring that up, I was like, Chris, my guy, amyloid world for the win. Yeah, so I was really excited to see you bring up that paper. It's an excellent review. I knew the exact one you were talking about.
It's all theoretical, obviously.
Yeah.
But I mean, I don't know. I find it particularly persuasive.
Yeah, I find it very compelling, especially because of how it solves the chirality problem, which I find, and then also the fact that like peptides will naturally form, like so it's
And
not like a
and
whole
the lack of
functional
RNA
protein or whatever,
and the
but
issue around RNA stability too.
yeah.
Right? Yeah.
And so I find it particularly compelling, not only for that, but because like the peptide can help catalyze like its own reaction to create more amyloids.
Right.
And it can pass on. It has some level of inheritance, like because the scaffold can reflect like environmental information, or like the scaffold will create daughter scaffolds that are similar in composition to it. So it sort of like shows an ability to pass on pass on information generation to generation in a way that's like inheritance without
It's
DNA.
like inheritance. Yes.
So it's like,
and that it can vary.
Yeah.
Yes.
And yeah, it can vary, it can have mutations, it can have mistakes, which is like everything that is necessary for life. So I find it very compelling as well.
Same.
Like
I feel the same way.
Because I definitely think that like regardless of whether you have replicator first abiogenesis or metabolism first, it seems most logical that you would have to start, or at least very early on in the process, you would have to have like peptides.
Yes.
Like,
it makes the most sense to me. I mean, given the
before
stability,
you would have DNA or RNA, I can't really see you having DNA or RNA without having peptides first.
Right. I agree.
And whether like I know that there are some other theories about like lipid first abiogenesis, which is interesting.
I'm not as familiar with the ideas behind that. I've read into amyloid and RNA world.
The lipid first is really interesting because basically they posit like you have the same kind of inheritability. Basically, like if you imagine like a lipid that's composed of like different kinds of like chemicals within like the membrane, not just all the same type of lipid. But when the lipid divides, right, say that there's like, you know, like the lipid gets too big, or there's some kind of disturbance in the environment and the lipid like divides, there's going to be a difference between each of the daughter cells based on just what was the composition of the membrane when it split, because the membrane would not be uniform. It would have like different identities of chemicals on one side versus the other. So when it splits, you have two daughter cells that resemble the same composition as the parental like lipid bilayer, but have slightly different combinations, and their daughters like will have slightly different. So it's like kind of a way of having like inheritability, replication, those kinds of almost Darwinian kind of processes, but occurring without DNA or even without peptides.
Sure.
So it's also pretty interesting as sort of like
and gives it almost a scale-free way of it happening, because you're talking about, you know, potentially vacuoles or bilayers that can be various sizes.
All you need is some sort of like environmental process that is like creating these like lipid bilayer bubbles that would, and you would have some sort of disturbance, whether that's from like moving water or air bubbles or whatever is a mechanical disturbance that would cause these lipid bubbles to split off.
Interesting.
So that there's lots of, I mean, I just love abiogenesis. So I think it's such an interesting, compelling concept.
Right.
So
and
then one thing that a lot of these theories kind of leave out is just sort of like the minerals or the metals that would be in the environment at the time. And it's super interesting and compelling stuff. People tend to discount the atmospheric composition of the early Earth would have had like very, very little oxygen and a lot more carbon dioxide, which people don't really think, and I guess unless you're like an environmental chemist or whatever, but that totally changes the ocean chemistry.
Oh, for sure.
So that allows for a lot more nitrogen and iron to build up in the water. With that lack of oxygen, with all that carbon dioxide, and you have a lot more.
With that additional nitrogen, that's just going to be a crucible for peptigenesis and other organic compounds that are going to be critical for early life.
And it's a
That-
huge reducing environment too. It, like, as
Mm.
opposed to like an oxidizing
yes,
environment, like
right, without
today.
any oxygen.
Yeah, it's a
Right.
totally different chemical environment. So like the kinds of chemistry that we see in the oceans today just totally do not apply to an early Earth.
Right.
Which lots of people tend to discount, especially creationists almost never bring that up. So
For sure.
yeah, I mean, I hear abiogenesis coming up all the time in
Oh,
these
sure.
creationist circles.
They
I'm
love to talk about it because it's a
Yeah.
place where we have a lot of ideas and a lot of theories and a lot of models, but no strong consensus. And so, of course, they're going to bring it up.
And it's an area that they think that they can win no matter what. Because if you bring up, like, even if we had an experiment where like actual life was created in a test tube, they would just say, "Oh, see, it requires intelligence."
Exactly. Of course, they're going to do that. Yes. Right. I mean, if you hear the way if you go back and listen to the beginning of this, the way Pitch Lumin, Nathan was talking about this issue. I mean, it's essentially, he was saying, like, I would be willing to accept it if you could set up conditions, but as long as it matched closely like actual natural conditions. But you can tell that there's this opening of like, but it has to be absolutely natural. So if there's any sort of exception where we did it in a beaker or we did it in a lab, now all of a sudden they can say, well, obviously, look, like, yeah, an intelligently designed and intelligently created life, right? That's proof that intelligent design occurred.
Yeah. And then meanwhile, you have like absolutely no skepticism that a
Right.
mind could exist without a brain. Like they're totally fine with that. I mean, that no question.
Yes, as I was arguing with Refined Living on your channel. I don't know if you looked through that, but
Oh God, well, that
don't
that's a whole
look.
can of worms with that.
Yeah. God. The presupposition thing. I don't know if Amiga Nuts is still here. Do birds have the equivalence of a human TKTL1 gene? I would assume. I don't know if you're bringing that up because I brought that up with Jamie and Nathan in our after show. That's when I discussed it most recently. I don't know if you know anything about the TKTL1 gene. Grayson, have you heard of this?
I actually haven't. I was just reading the wiki right now.
So it's one thing I think is particularly interesting about it is that it is one of the few genes that when you compare the ancient genome of Neanderthals to humans and then also to chimpanzees, that Neanderthals have a chimpanzee version of the TKTL1 gene, and humans obviously are the distinct outlier there. So most genes, when you compare the genome between humans, chimps, and Neanderthals, they're going to be mostly like similar between Neanderthals and humans, and then the chimps are the outlier.
Mhm.
But there's a handful of these genes that are not. And there's this really cool paper that was published out of Svante Pääbo's lab just this last year, showing a single mutation in the TKTL1 gene led to a single amino acid substitution in humans. And they did some experiments even with mice, introducing the human version where they were able to increase neurogenesis in the parts of the brain that feeds into the frontal and prefrontal cortex, so that the mice actually have larger prefrontal and frontal cortices with the human version. But if they put in the ape version, they don't get that change.
That's really interesting.
A single amino acid substitution.
Yeah, so that's really interesting because to my knowledge, Neanderthals had larger overall brain case size than humans. But I guess the question would be which parts of their brain was bigger.
And actually, the we know that their frontal and prefrontal cortices were relatively small compared to their overall brain size.
Yeah, so then their executive functioning would have been.
I mean, I would imagine it's still pretty good. But yes, that's one suggestion that obviously, like with modern humans, if we have even more frontal and prefrontal cortex brain space, that would be higher.
Well, this is a very good example because Standing for Truth, Donnie loves to bring up Neanderthals. He wanted to talk to me about Neanderthals because he thinks that Neanderthals are obviously like degenerate humans. So his creationist worldview would posit that sometime either after the flood or after the Tower of Babel, that humans would have spread out across the earth and would degenerate
Right.
into all of the different humans we have today. And so in that worldview, like the Neanderthals would be derived from humans,
Right.
which is
Yes.
obviously like not the current scientific understanding.
Obviously not.
That like,
Yes.
yeah, we share a common ancestor with
Mhm.
Neanderthals, but Neanderthals did not evolve from humans.
Right.
So this is a kind of
And they
nice little
use
line
that.
of evidence.
They say that part of the reason why they have to do that is because the genetic evidence clearly shows that Northern Europeans have a a pretty substantial
Yeah.
percentage of their DNA that's Neanderthal in nature,
Yeah.
relative to say Africans, especially Africans. And then of course you have the Asians with the Denisovians
Yeah.
DNA, right? So the way that they try to explain that is that difference arose from the three sons of Noah,
Yeah.
and that
Yeah.
Japheth, gave rise to the Northern Europeans, and that therefore, like, the Neanderthals are descendants of Japheth, and that yes, like that there was degeneration that occurred to the point where then you had this separate group of Neanderthals that were degenerate in
Yeah.
some way.
What
But
I
they're
find
it's
very
just so crazy.
fascinating about that position is is how they can explain things like are you familiar with Homo georgicus?
No.
That's one of my favorite examples because, well, they just don't know about it. It's like not that many people outside of like paleoanthropology really do. It's kind of like a lesser known, but it's considered a subcategory of Homo erectus, like a very,
Okay.
very early version of Homo erectus. But when you look at like the skulls, they look so ape-like and they're so diminutive. They're so small in size
Oh.
and they're so ape-like. But the thing is that they're in Georgia, like the country of Georgia. That's why they're Homo
Oh.
georgicus,
Okay.
which is right next to where they think Noah's Ark landed in Turkey. So they're essentially saying that I get if Donnie says, oh, all these humans went out to the far corners, the remote areas of the earth, and they became Neanderthals, or they became Denisovans, or they became Homo floresiensis in the Philippine Islands or whatever. But this is right in the backyard of where the supposed ark is supposed to have landed.
Right.
It's not like this would have been a very remote population for like
Clearly
humans in the
not.
Bronze Age or whatever. Like,
Mhm.
there's no way that you would have this kind of degeneration to like make something that looks so different, like Homo georgicus.
Of
Like,
course.
it
Right.
makes no sense.
And the fact that you could have like Neanderthals emerge, uh, have their DNA be so distinct that they
Yeah,
clearly are a separate group.
they cluster completely
The
separately.
cluster completely separately, but that all happened within the last 4,000 years at
Yeah.
post flood. Just insane.
Well,
It
the
wouldn't
thing
make any
is,
they
I've
would
heard them
cluster so
say that,
separately.
oh,
Yeah,
what if Neanderthal is a super mutator?
right.
So
But
they're a
then why are
super
the
mutator.
Denisovans and the Neanderthals so similar to each other? Because that's
But
the other
what
thing
I
that you have to
ask
take
them
into
about
account.
that is, well, okay, so on the one hand, you say that they're super mutators, and on the other hand, you admit that they're incredibly genetically homozygous.
Right.
So those two things don't add up.
They
Like,
are
you can't
genetically
be genetically
homozygous.
homozygous and a super
Yes.
mutator.
Well, and like they cluster. So there's another. So this is a slide that I have where I'm showing like the chimp version to the human version for the TKTL1, showing
Mhm.
that one single amino acid difference. But I have another one where it was a conversation with Sal Cordova.
Oh, lovely.
I've talked with him a few times.
Yeah.
Yeah.
He's told me that I don't even go to the fossils. I don't talk about any
Well,
fossils.
yeah, he doesn't. I know. Of course he doesn't.
He doesn't want to acknowledge the fossils.
No, of course not.
And I think that he would just assume that Australopithecus is a real creature that's just like a separate creation of God that was like a bipedal ape, missing link,
Right.
exactly predicted by evolution.
So when you compare mitochondrial, you do like pairwise sequence differences between humans and Neanderthals and chimps. Like humans to any human, there's going to be more similarity in mitochondrial DNA, right? Of course, like with the existing human mitochondrial sequences. And so like any one human compared to another, it might be as little as just one difference, or it could be as much as around like 15 or so differences. but the average being about 8 or so of this particular like sequence within the mitochondrial genome. But when you compare humans to Neanderthals, you know, they're different. They're non-overlapping. And like you can see it clusters separately, as does like humans to chimps. And of course, chimps are like more than twice different than humans to Neanderthals. And then, you know, we can find sequences, of course, with Neanderthal genomes doing the same thing, mitochondrial phylogeny, chimps and bonobos being very separate and distinct, right? This is the phylogenetic tree. When you zoom in, we can see like, you know, African populations segregating out from non-Africans with a single sort of monophyly with a couple of different subgroups. But then here's the Neanderthals, and you can see that they're different, and they don't cluster at all with humans. They're completely distinct. And then this is what I was talking about with the TKTL1 gene and showing
I really, really hope that within my lifetime we get to find the genome for like Homo erectus, or
I would love that.
even
so
like
much.
Australopithecus, which would
Yes.
probably not be very possible, but that would be like the dream. That would be so cool.
We keep pushing it further and further. I mean, the hope with Homo erectus is that we can find some Homo erectus specimen that somehow made its way into the last several hundreds of thousands of years and is also in a location that's relatively cold. So we mostly get ancient genomes from like cave sites where it's
so here's what I thought was one of the coolest like finds in all of ancient DNA, of like hominid DNA, for like the last decade, which has been like the whole burgeoning of the field of ancient DNA. The coolest find, in my opinion, right? So we found like genetic sequences for Denisovans, right? We were able to like sequence part of their genome. The coolest find was that, I think this was maybe in Tibet, I want to say, some part of China, but maybe I'm getting the location off. But they found a fragment of a jawbone. And at first, like, you know, I mean, they had not found any Denisovan bones outside of teeth. I believe that their DNA was
initially just teeth.
Yeah, it was just teeth. So they didn't have any bones. So they weren't sure what type of what type of species this fossil hominid jawbone belonged to. But what they were able to find was not DNA in the jawbone, but they actually found a type of protein in the jawbone, like somewhere within the jawbone. And they were able to show that this is not a human protein. This is not a Neanderthal protein, but they had the sequence of the gene in the Denisovan genome that codes for that protein.
That's cool. So they did mass spec to identify.
Cool.
Yeah. And that's actually one thing I think where ancient, not genomes, but ancient proteomes has a lot of potential. Because, you know, you hear creationists talk about how there's soft tissue being found in dinosaur fossils.
Oh God.
I know. And they don't understand what that means. They think what it means is that like you break open a dinosaur fossil and like blood pours out or something like that. And clearly that's not what it is. And when you learn the details, it actually becomes really fascinating. A few things. One, like it shows that there's an extraordinary long half-life to some proteins. But it's only a handful of proteins. Like they were only able to extract like 6 proteins from dinosaur fossils. But I think what I don't know, and I don't know if this has been done, but I could imagine we could at least start doing this with many Cenozoic fossils, is that there's going to be some proteins that are going to have an even longer half-life than DNA does, and that we could extract the sequence from those proteins inside fossils. And that you can sort of get a retrofit. You're obviously limited. You can't get quite the genome data from that, but at least you could do codon, like you could because you're going to be missing the codon. You can get like you can narrow it down to certain codons for certain amino acids, but at the very least you can compare across proteins, and that would be great to do. And I have no idea about the feasibility of that, if that's just a pipe dream. But
ancient proteomics,
ancient proteomics, I think maybe that'll be the next big thing.
Yeah, that sounds really, really fascinating.
That would be great. Yeah, I mean, DNA has a very good, strong, like long half-life, and a lot of proteins do degrade faster than DNA does.
Thing, actually, just last year in 2022, they found the new longest preserved DNA. Did you hear about this in Greenland?
Yes, they did. Yes, like going back 2 million
It
years or
was 2
something.
million years.
Yeah.
Yeah, I think it was fragments from a bunch of different
A
animals,
bunch
but
of different
specifically
animals.
horseshoe
Just
crabs.
looking in the soil, like
Yes.
they were extracting from the soil.
It
That's
was
really awesome.
extremely iron-rich
Ah,
soil in
so
a
that's what it was.
very low moisture
Mhm.
environment in
Right.
cold
Cold.
Greenland.
Yeah.
And those combinations of environmental factors, were what's believed to be at play in the
Yeah.
long-term preservation, because, like, iron is, like, promotes cross-linking in a lot of organic
Mm.
materials. So sometimes I hear creationists go, look up the half-life for collagen. Okay, if you just look up the half-life and calculate it, you can see that clearly it can't exist in dinosaurs. And I'll be like, okay, well, like, first of all, like, the half-life for collagen only makes sense in a given environmental
Given
condition.
environmental condition.
Like,
Yes, exactly.
if you change the environmental condition, obviously you're gonna change the preservation of collagen, like,
Of
that's
course.
obvious.
Right.
And then the second thing is that that's not cross-linked collagen. If you cross-link collagen, you are changing its chemical
Mhm.
structure and increasing its half-life by loads. I
Sure.
mean, you're making it a much more stable compound. If you're cross-linking it, you're structurally reinforcing it, in ways that normal collagen wouldn't have. So you're going to preserve it exponentially longer in that case as well. And
Sure.
iron promotes this cross-linking. So yeah, there's numerous things that are wrong with their little
Of course. Right.
breakdown.
Right.
But yeah, the number one thing is, are you aware of Dr. Fuzz Rana?
Oh, yeah.
He's an old Earth creationist,
Mhm.
but he actually has a degree in biochemistry.
Mhm.
And when he talks about these soft tissues, these so-called soft tissues, I wouldn't even really call them soft tissues, but these soft tissues in dinosaur bones, he's like, okay, yeah, this is proof that the Earth cannot be young. Because when you actually look at what you're looking at, what you're finding in those bones is clearly, like, highly cross-linked, highly degenerated, like, of versions of only the most stable organic molecules,
Exactly.
which is exactly what we would not find if these dinosaur bones were like 6,000 years or
Yeah.
something.
Or 4,000 years, right? Because they were in the flood.
Yeah, yeah.
Yeah.
So, like, we would not be finding these organic molecules
And
in these
exactly.
kinds of states that we
Right.
are seeing. That would
And
not be occurring.
you can find, right, there are all kinds of fossil specimens that you can find in, say, Antarctica, or even in parts of, like, Greenland, or in parts of Asia that are very, very cold. Especially Antarctica, like those are going to be organisms that lived, like the ones that they're pulling out there are 30 million years old, 40 million years old, or
Mhm.
even older, and sometimes hundreds of millions of years old. Why can't we get DNA from those specimens if they're only 4,000 years old, according to creationists? Those are ideal conditions for DNA preservation, like being frozen.
The funniest part was, I had a debate with T Jump about the age of the Earth, and this subject came up, and I said, how come we never even find even fragments of dinosaur DNA?
Mhm.
And he said, "Oh, but we do. We found a few letters of dinosaur DNA." I'm like, "What? Where are you reading this?"
Yeah, where is
Like,
that?
I've tried to find his source on this,
I
and
think
I have found nothing. And, like, if they were to say, like, "Oh, well, we found an ACT sequence, like, of, like, 3 or 4 base pairs long," I'd be like, how could
man,
you possibly know that that's dinosaur? Like, how could you
of
know that that's
course.
not just, like, if it's such a small fragment, like, how do you know that's not just, like, bacterial contamination from, like, because I am aware of a story where they found DNA in dinosaur fossils, and they were like, "Huh, that's really weird," and they decided to sequence it, and they found out that it was a bacteria genome.
Oh,
And they're
shocking.
like, "Oh, well, yeah, that explains that. A bunch of bacteria migrated into these fossils," which
Sure.
is a
Yeah,
common enough occurrence.
of course. Yeah, the world's covered with microbes. Yeah,
Yeah. So
right.
I would That's the only thing that I could even think that he could be referencing. But when I
Right.
tried to bring up the point that we do not find dinosaur DNA, which we very much would if they were only 4,000 years old.
Yes.
And he was like, "Oh yeah, no, we do have dinosaur DNA." I was like,
No.
"Yeah, what fantasy world are
Mhm.
these people living in?"
Or what about older than dinosaurs? I mean, they're all 4,000 years old according to their model.
Yeah, great
Like, why
point.
can't we get trilobite DNA?
Yeah. And
Right?
I've asked, well, the fossil record is always such a hilarious topic to bring up with creationists
The
in general,
hydrological
about like the
sorting.
ordering of the fossil
I
record. But
know.
yeah, I
It's
mean
so much wish casting.
it
It really is.
is patently absurd, some of the things that I have heard to try to explain how you can get the order of the fossil record that we observe is like
Yeah.
the most ludicrous like imaginings that
Mhm.
I've ever heard about how physics, how the world works.
I
It,
know.
it just is.
Like
It boggles me.
Hovind, I hadn't heard this model before, but he has this idea that there's like this tidal wave that's just crashing around the earth, and that's why
Yeah.
we get the layers. And but then eventually it stopped. I mean, he doesn't ever provide any specifics.
I don't know if you
Um,
saw the video that I did
mhm.
with Kent, where I asked him that specifically. I was asking about like the Grand Canyon and how we find layers that are intermediate within the Grand Canyon, where there's like animal tracks, or there's
Oh,
burrows,
right.
or things that are like if these were all laid down simultaneously by a flood, you could not get animal footprints on on a layer in the middle of that.
Yes,
And he
right.
says, "Oh, well, during the flood year, the there were certain areas that were in the flood and they dried out as the flood moved
Exactly.
over to different areas and animals walked over them, and then the flood came back and deposited new layers on top of that." And I'm like, "What is this game of like global whack-a-mole with the
It totally
flood?"
is. Yes,
Like,
and it's just this I mean, it really is just wish casting. It's this like I'm going to just come up with any kind of goofy way of just explaining this thing, and I'm never ever going to have to address it again because you're going to be the only person who will ever talk to me about this. And the next time I'm in front of some gullible audience, I'm just going to give them the same spiel that I always do.
yep.
And that's exactly what it is. I mean, you can find examples like that all the time, but one of the most fascinating ones is with the Ediacaran biota in
Yeah.
Australia. So like there we know about their overall like the population and like the diversity of animals in the Ediacaran,
Mhm.
most from the Australian assemblage. And the thing that they do about that is that they basically are taking layer and after layer, and they have square meters that they take from, and they have tracks of the animals. They'll find feeding stations. So like Dickinsonia will move along and then stop and settle in, and they'll know that that animal did that for a while because it's like settling in and depressing, and then
Mm.
it moves a little bit more, and then they see the same animal again, and they can track this for meters and meters. So then they take that layer off, and then they have another layer below with different animals doing the same exact thing. How could
Oh,
that
yeah, interesting.
possibly? And they have layers and layers and layers and layers of this. And you're going to tell me that like each of those layers was just because like a wave came by, and that like then it was like free to settle, and then these animals were feeding for a little while, and then another tidal wave came over and then layered it over again, and then the animals are feeding on that for a while, and then all of this happened within like a few months.
Yeah.
Come on.
My
It's so
favorite thing to bring
stupid.
up is that's really good though, specifically with the Ediacaran
Yeah.
fossil tracks. But I like to bring up the animal tracks or like the different burrows or
Mhm.
something. Like you can't really have that if
Right.
if these layers were just like
And
being
we can
deposited by a
tell
flood.
exactly. We can tell from this rock that it was highly organic, and that like it was basically muck, and that the animal, the Dickinsonia, were in the muck and they were eating the muck. And so then they sunk in, and that's why you have these depressions that match that exact organism. And then we can
My
track that.
favorite point to bring up is because without fail, every single time that a creationist tries to explain the ordering of the fossil record into the various layers, they always bring up either like sorting based on body densities,
Mhm,
sorting based on mobility, sorting based on habitat, or
mhm.
sorting based on like intelligence, like some
Intelligence.
kind of
Right,
combination of those factors or
right.
all of those factors.
There's a reason why the mammals are up at the top, because they're the
Yeah.
most intelligent.
So my favorite point to bring up to this is like plankton.
Yes.
Like diatoms,
Diatoms.
foraminiferans,
Yep.
like the different types of like phyto and zoological like plankton that like are segregated in the fossil record. We can see that there are patterns of which types of diatoms are found in which layers, where foraminiferans are formed, like what type of plankton is in each layer is different.
Right.
But they all have the exact same like body density. They all have the same intelligence. They all have the same mobility.
Which is
they
no
all
intelligence.
Have the same
Yeah.
like habitat. Like everything is the same.
Right. And that if I correct me if I'm wrong, but there's like the diatoms didn't really come on board, and I mean, like you don't
It's not
find
until like the Jurassic or like the
yes,
Mesozoic
exactly
somewhere.
right.
Yeah.
You don't find any Paleozoic diatoms.
Yeah.
So like, wouldn't they have sunk? Like, why they're not any? I mean, obviously they're not intelligent. That's not why.
Yeah, you would find them if there were both diatoms and
Right.
foraminiferans in the same environment, then they would absolutely be fossilizing together based on any known physical mechanism of fossilization. Like there's nothing that would fossilize foraminiferans but would not fossilize diatoms. So like the fact that they're not found in the same layers, like the diatoms are only found in the upper layers, never the lower level layers. There's no physical explanation for how that could be possible besides an evolutionary like model with like deep time and the scientific
the same kind of argument can be made from looking at the diversity and change that we see in other invertebrates, particularly in the Paleozoic, but extending past that too. So one example are the trilobites, which we can date. Like you can make a very good estimate of precisely which, how old a rock is based on the kind of trilobite that you can find, because of like the change that we observe over time. But another great example of that is with brachiopods. So brachiopods were extraordinarily diverse through the Paleozoic, particularly like Ordovician, Silurian, but we still have them today, and the diversity and the way that they look changes so much over time, and they all order across time. Like, why can you not find Ordovician brachiopods today, despite the fact that we have still brachiopods that are very different from the ones from the Ordovician?
And then the last thing I want to bring up with like the plankton, because I really this is the most hilarious point to me, is that if you were to take like all of the diatoms in our fossil record, all of the foraminiferans, all of the various types of plankton that we have fossilized, like all the diatomaceous earth, right? If all of those plankton came from the same event, like a flood, and they were all alive at the same time during the flood, even if you had water covering the entire surface of the earth, there would be so many plankton alive at the same time that like it would be like
it would choke the oceans.
The water would be like a sludge. Like it would not even flow like water. It would be so,
and if they all died,
choked with plankton.
The toxicity and I think the heat generated from them rotting would like boil the ocean.
Yeah, it would kill everything. I mean, nothing would be able to survive if the waters were that choked with plankton.
There's so much that just is completely precluding their model. Oh God, it's so stupid.
Yeah. I mean, I feel like if anyone thinks about this for more than 2 minutes, like, then there's no way they could believe it. But like, then you have people that dedicate their entire lives to trying to research this stuff, and they still like. It blows my mind, like how Nathaniel Jeanson can have a PhD in genetics from Harvard, and write a book like he wrote.
Yeah, or Sanford, same thing.
Yeah.
Right. Yeah.
Well, I think ultimately those types have to know that they're full of it, and they have to be nefarious, because at that level of education, I mean, you know better.
Of course, yes. I mean, I can't imagine that they're that delusional. Yeah, that it has to be in bad faith.
I will say, I've worked with many PhDs and MDs, and I am a firm believer that intelligence is compartmentalized. Like you can be a genius about a certain subject, a little niche, and and you can be an absolute buffoon in other areas where you're
Yep.
like, you're just not directing the same level of scrutiny or intellectual analysis in other areas of your life. They're like blind spots.
It's a blind spot. Absolutely. I've met professional like biologists, a handful, in my professional capacity. So these aren't like active creationists, but they believe in creationism.
Wow.
And they won't ever talk about the details because I think they know like how ridiculous it really is. But it's a blind spot, and they just will focus on their thing, and they're going to ignore it. But if it ever comes up, then they might argue and think like that there's a different way of thinking about it. But and the other thing is that like so many actual scientists, like biologists, who of course accept evolution, I mean, they don't think about evolution as deeply as like you and I do, unless they actually work on it, for the most part. Yeah, that's a good point.
Well, during 2020, I knew a specific orthopedic surgeon, so an MD, who would flat out tell you that COVID is nothing any different than the flu.
Right.
And he would say, "Oh yeah, you know, Trump watermarked all of the ballots, and he's going to show the election fraud, and he's going to be the president." And I heard all these lines from an MD,
Sure,
like
an intelligent person.
Yeah.
Yes.
So absolutely, I believe intelligence is compartmentalized. Some
Very
of the
much so.
smartest people I know about in some areas are some of the dumbest in others.
Yep.
And yeah, James Tour is a wonderful example of that. I don't know if you watch his videos.
I've seen Professor Dave Explains Tear 'em Up, which was fun. Yeah.
So I've had a very limited correspondence with Dr. Tour, but in one of those videos that he made as a response to Professor Dave, he was asking for a paper. And this is all going to tie in with the amyloid world hypothesis, actually, because he was asking for a paper. I don't remember the exact specifics, but he like put out a challenge. Somebody provide me a paper that shows X. And then he was like, look, none of these professionals could do it. I asked them in a Zoom call and they couldn't do it. Dave's never done it. And I was like, oh, well, like that paper's right here. Like, and I sent him the paper and I was like, hey, this is the paper, the exact paper that you asked for. I think he was asking for, like proteins forming from amino acids or something like that. I think in an aqueous environment or but like I literally sent him I made sure like
the paper
every single criteria that he asked for. This paper checked it all off. It was within the context of the amyloid world hypothesis. It was by one of the same scientists that is in that area. And I was like, hey, here's the exact paper. And he says, well, those don't show all of the amino acids they formed. They only used certain amino acids and they didn't show all 20, but they did show what I asked for, like with 4 amino acids. I'm like, yeah, well, you didn't specify. And he's like, but regardless, I get so many emails. I can't possibly make a video on every paper that somebody sends me. And then like he never brings it up again. And obviously he's going to continue to ignore the existence of this paper.
Yep.
And he wouldn't
never refer to that correspondence, and it'll just go down the memory hole.
Yeah. I would really love to debate James Tour on abiogenesis one day.
I would love to see that.
It's one of my favorite topics. I mean, I've been trying to debate Otangelo about it, and he won't even talk to me. So,
well, I wouldn't want to do that.
Yeah, well, he's a bit of one of those, like he's like kind of like a crackpot that's like written entire like novel length of material on abiogenesis.
And it's like, what is this stuff? Oh my God.
It's all nonsensical,
Yeah.
One of my favorite things has been the recent advent of creationists talking with ChatGPT.
Oh, really?
I don't know if you've seen any of this at all, but it's absolutely hilarious. Either they lie and say, here, this is what I said to ChatGPT. And then they're like, and here's what ChatGPT said in response. And it's like, that is correct. The Big Bang is a lie and the creation happened. And obviously intelligent design is necessary for genetics. And I'm like, that's such BS. And then I have, I'll just copy what they said to ChatGPT and paste. And then it's literally like ChatGPT is saying the opposite of what they say. But I really, really want, like my goal for the next couple of months is to try to get a debate of like me, a creationist, versus ChatGPT. So like the three of us in conversation where like ChatGPT can kind of be a fact-checking moderator.
Okay, don't do that.
Why not?
Let me tell you why. All right, and maybe it'll change, but I did something with ChatGPT too as well. I'm going to zoom in here so we can actually read it. Ooh, a little too much. Hey, look, there's my little icon there. All right, so I was working on a grant, and I was trying to. I was like, okay, I don't really want to work on my grant right now. I hit a little bit of a wall, and I was trying to get something addressed. So I was like, well, let me check out this ChatGPT thing and see if it can like address this. So basically, I asked so my grant was on like, how does lead poisoning affect how thyroid hormone works in the brain? So I was like, well, let's see what ChatGPT comes up with. And it came up with an answer that is like really cogent and seems like it has a lot of ideas, much of which is probably true. Like we certainly know that lead poisoning can affect thyroid hormone function in the brain by affecting transport and metabolism of thyroid hormones. Like that's true. So then there's a particular thing. I can kind of skip ahead. And unfortunately, one of the questions got cut out, but I asked it, how does lead affect how transthyretin works, which is a protein that plays a role in the transport of thyroid hormones in the blood and the brain. Lead can bind to transthyretin and alter its function, reducing its ability to bind and transport thyroid hormones. Okay, this can result in lower levels of thyroid hormone in the brain and reduce the availability of hormones for their normal functions. All right, so already this part here, like this is a key part of what my grant was about. I was looking at like how does lead impair the production of transthyretin in the brain, which is a carrier of thyroid hormone in the brain. And it says that lead can bind to transthyretin and alter its function, reducing its ability to bind and transport thyroid hormones. Okay, I don't know of any paper that's shown that. So then I asked, what is a study that shows that lead can bind to transthyretin? And it said one study that demonstrates this, that lead can bind to transthyretin, is a study published in the journal Toxicology and Applied Pharmacology in 2000. The study, titled Lead Binding Properties of Human Transthyretin, found that lead can bind transthyretin in vitro and in vivo, altering its structure and reducing its ability to transport thyroid hormones. Great. And then it goes on to explain like the conclusions. And I'm like, all right, well, this is a study I definitely need to know. But I wanted to find a little bit more information. So like, who is the author of the study titled Lead Binding Properties of Human Transthyretin? The authors of the study titled blah, blah, blah are J.M. Moore, J.L. Boyer, and M.J. Cosnett. It was published in Toxicology and Applied Pharmacology in 2000. So I go to the journal, the Toxicology and Applied Pharmacology, and I look for this study, and I can't find it. It's not there.
Whoa.
There's nothing there. I then Google this phrase, lead binding properties of human transthyretin. Nothing shows up when I put it in quotes, not a single thing. When I just Google the phrase without it in quotes, I get ligand binding properties of human transthyretin, which is very different from lead binding properties of human transthyretin. And the authors are not these guys. And it was not published in Toxicology and Applied Pharmacology in 2000.
That is so weird.
It made up an entire reference.
Whoa.
And it made up the justification for that reference and lied to me about an actual reference that does not even exist.
That is definitely, yeah, that's worse than anything I've experienced with it. I have experienced holes or inconsistencies in its knowledge before, but never anything like that. Wow.
And no one, I mean, this explanation it gave is perfectly reasonable. It seems like it would be fine. Like I could buy this. Like, and I was very intrigued because, okay, Jesus, I'm trying to get money from the government so that I can address some of these questions to try to figure it out. I'm trying to work on something that I think no one has done. So if someone has shown that lead can bind transthyretin and alter its function, reducing its ability to bind and transport thyroid hormones in the brain or wherever, like I would definitely want to know that. When I asked for the study, it gave me a citation.
You're right. It's nowhere on Google Scholar, but I am finding like ligand binding properties of transthyretin. But you're right. Different authors, different year.
different year. It's 2001, I think, is when that one was published.
I see 2011, but
or maybe yes, it's 2011. That's right.
twos and ones.
But that paper, ligand binding properties of human transthyretin, all that means ligand is just simply
just
like,
anything that binds to an enzyme.
Exactly. Yeah, it has nothing to do with lead, and there's no
that's
lead mentioned in that
ridiculous.
paper.
Wow.
Mhm.
That is crazy. So the worst that I've experienced with ChatGPT was like I like using it for like asking questions that I would normally ask to Google. And so I've been doing like research for some of my videos. I wanted to do a video on like ancient atheists in India.
Mhm.
And so I was curious, like, were these the first atheists recorded in history? And so I asked ChatGPT, what were the earliest atheists in recorded history? And it gave me a bunch of ancient Greeks from years after the ancient Indian atheists that I was talking about. So I was like, well, ChatGPT, how about these guys? And I asked ChatGPT, and they're like, oh, correct. These are like, some of the earliest atheists in history from 200 years prior to the ones I just told you were the earliest known ones in history. So I'm like, okay, well, you do know about these ancient atheists in India,
Mhm.
but when I asked you what was the earliest known, you only gave me Greeks. So what's up with that? So it definitely, like, there was some sort of like inconsistency, or like, because
Mhm.
it seemed to have knowledge about the ancient
Yes.
Indian atheists that I
Right.
asked it about, but it was
Right.
only after I knew to ask it.
So I don't think that it has like synthesis of knowledge. Like it has these lines of thought and things that it can draw on, and it can string together novel sentences that sound like someone is actually writing you and explaining this to you. But it is just stuff that's just grabbed, that some of it's going to be true and some of it's going to be accurate and some of it's not. And
Yeah.
some of it's just making up.
Very difficult to parse what's what.
Yes.
But to my knowledge,
Nice.
though, those the ancient Indians that I'm going to be covering in my video were the first known, or like the first recorded, like documented atheists in history, which is pretty interesting.
That's cool.
Because most people think about like ancient Greek philosophers or stuff
Right.
like that, but the Indians had them beat by 100 or 200 years or so.
Oh, that's really interesting. Yeah. I mean, and we, as Western minded folks, unfortunately, ignore so much that happens
Yeah.
outside of
The most
Western thought.
fascinating thing, and I actually sent this to my friend of mine that is who teaches like intro to philosophy on like the undergraduate level, and I was like, "Hey, were you aware of this?" Because years ago when we were in college, he first taught me about Hume's problem of induction,
Mhm.
and it turns out that the exact problem of induction was well known and described by ancient Indian philosophers, like in like 500 BC or before. So
Wow.
it's like Hume was not the first dude to come up with this. He got all the credit. It's Hume's problem of induction, but it was known to ancient Indian philosophers like over 2000 years before Hume. So
Wow.
just pretty fascinating stuff.
That's amazing. Yeah.
Yeah, there's actually like there's a ton of stuff that I was learning about some of these ancient Indian philosophies. It's like they're not Hindu. It's part of this like Sramana movement.
Okay.
But it's really fascinating. There were a bunch of different philosophies. This is like around the environment that Buddhism came out of. So
Okay.
like the Buddha was living in like where there were all of these different schools and philosophies in this kingdom where like the Buddha was a part of, and they believed all sorts of different things. There were the agnostics, there were the materialists, there were the people that believed in karma but no gods, and like all kinds of like interesting philosophies that were all kind of existing in the same like area and time period. As like that's where Jainism comes from, is that kind of Sramana movement. Buddhism comes out of that movement. Like, but there were a lot of atheist traditions that kind of like died out. Like there are no modern descendants to those philosophical schools, but they still existed.
That's very cool. Huh. I had no idea. I would not have guessed that at all. I'm looking
Yeah,
forward to
exactly.
it.
It's like
Seeing that.
very little people in the West or even in India really know
Sure.
or learn about this because, like, you know, like with Hindu nationalism on the rise in
Of
India so
course.
much, they don't really learn about a
Yeah.
bunch of
I mean,
atheistic
just as like
philosophers.
the tradition of atheism in the United States is completely washed
Yeah.
away. You know, the fact that it was like so prominent in the early 20th century, like in the Midwest of all
Yeah.
places.
Or I mean,
So,
freaking right from the beginning with Thomas
yeah,
Jefferson.
that
I
as
mean,
well.
he's a deist, but
Right.
still, I mean,
Still, right. It's as
you
much
talk
of atheist as you could be at that time.
yeah,
So,
look at his
yeah.
comments about Paul and tell me that man's a
Mhm.
Christian. I mean,
Yeah. I mean, he edited the Bible
yeah.
to talk about sacrilegious.
Yeah, exactly.
So,
I
all
mean, I'm
right.
not the greatest fan of Thomas Jefferson, but
No, there's
sometimes
a lot to
he
not
did
like
some
about
pretty
Thomas
cool stuff.
Jefferson, but he was at least interesting in many ways.
Yeah.
Yeah. I do need to run. I gotta
Yeah,
actually work.
sounds good. It was a
Yes.
fascinating
Awesome
discussion
talking
tonight.
with you. Yeah, this has been good. I was not anticipating a chat session with you tonight, but that's great. I'm
Yeah.
glad we did that.
And hey, you
That's
got to bust out some of your past research and talk about
yeah,
it.
I was
It
not
was super
expecting
cool.
that. That's crazy that you mentioned that.
Yeah, it was
Yeah,
super cool because
yeah.
again, I was a little bit fuzzy on some of the details for Fox P2.
It
So
was enough for me to pull that out. So
yeah, well, it was
cool.
cool to get a refresher on some of these
Right.
concepts as well. So
Yeah.
thanks for having the time to talk to me.
Yeah, no, this has been great. I am ending the broadcast. Thanks to everyone watching this. If you've made it this far, Amiga Nuts, thanks for participating in our little conversation here as well. And yeah, anyone watching this, please like, subscribe, all that stuff. If you made it to 3 hours and 4 minutes, so talk to you later. Bye, guys. We'll see you around, Grayson.
Peace out.
Bye, bye.